It's another Sunday. Unfortunately, we had a little bit of a technical issue last Sunday, but here we get a refresh of basically understanding a full week of digesting everything that we learned last week. And then we're going to talk real heavy today with our friends Leif-Olsen and Craig Trester. We appreciate you guys. This is a hand selected from Leighton Morrison, so you guys come highly respected and highly sought after. So we're excited to learn a lot from you guys. I want to give a shout out to our title sponsor, Agro Cocoa. We appreciate making everything that we're doing today cheaper for all of the students. So thank you, Agro Cocoa. Check them out at agro.cocoa on Instagram. In this session, sponsor is Blue Mats. So shout out to Michael and Steve over there at Blue Mats. We appreciate you guys. Thank you so much, Blue Mats, for sponsoring this session. And this session is yeast and saprophytic fungi in the soil. Taught by some of the best and brightest to do it. So guys, if you'd like to take a moment, kind of introduce yourselves and then kind of get into the presentation. And for you guys that are checking this out live, we're going to have a Q&A after the presentation. Take it away, guys.
Yeah. Thanks for tuning in, everyone, and joining us a week late instead of last Sunday. My name is Leif-Olsen. I'll give you a brief synopsis of why I talk about the stuff I do. I originally studied ecology, behavioral ecology, kind of like climate change, plant response stuff, and then transitioned from that into more of an environmental chemistry angle on things like biogeochemistry, bioremediation, how plants and other organisms can deal with pollutants, which then eventually led me more into the mycology world. Worked in mushroom cultivation for a little while and then transitioned into environmental consulting with a range of topics, but really I've always been really intrigued and fascinated with the roles that fungi play in the environment and then how they can be leveraged to improve things like our land management, our ability to grow plants, to restore landscapes and things of that nature.
All right. My name is Craig Trester. I am an applied mycologist, educator, and citizen scientist. I got into soil through the aspect, mostly through fungi. My interest in my study of fungi and mycology stemmed from an aspect on kind of bioremediation. Much of my study in university was kind of focusing on environmental policy and looking for alternatives which are leveraging the metabolic capacity to break down organic and inorganic contaminants and immobilize inorganic contaminants. And kind of heard about phytoremediation, remediation of plants, remediation with microbes, bacteria, algae. But then I heard about micromediation, but then realized how little I knew about fungi, mostly because even though I did study parts of biology in my education undergrad, there's this big kind of hole missing. So that kind of led down the track of learning about more of some of the applied mycology, learning about fungi and biology and soil, setting up a project community lab, teaching about fungi in New York City, and then eventually I head down that path into soil. So given the fact that fungi are ubiquitous and they are ultimately the architects of the soil and build that larger structure. So definitely today we want to talk about some of the important roles that fungi do play in the soil, obviously. Usually these are more talked about as the mycorrhizal fungi, the association that happens between plants and fungi. However, the fungi that play a huge role in the decomposition of organic matter, facilitating this nutrient cycling, are incredibly important. And also, too, certain roles of yeast in the soil, and aside from fermentation, which is kind of the cornerstone of our interaction with fungi as a species, but also the incredible protective and beneficial roles they can play, given the certain conditions facilitated by biodiversity and, a healthy ecology that is in place.
All right, so we'll get the slideshow started now. Let's get this going. And all right. Yeah, so saprobes and yeasts in soil, that's the topic of today's talk. It's not January 17th anymore, though. Forgot to update that. All right. So, we'll give you a little outline of what this talk is going to be like. We're going to start out by going a little in depth into the ecology of yeasts and other decomposer fungi. Then we'll talk about why these roles are important to plant health. Then we'll get a little more into the concept of ecological succession in the soil because it ties into all these topics. And then finally, we'll get into more practical hands-on management strategies that you can use for helping your soil fungi. And to start off, we're going to go through saprobes, yeasts, then talk about some of the ways that fungi convert energy and survive. And to start it off, I'll pass it back to Craig.
Definitely. So we're going to go a bit into the yeast life cycle here. Here's a nice little example kind of overview. When we talk about yeast, we're kind of most familiar as a human species with the yeast that bud. Now, yeast are a wide, diverse aspect of fungi. They're both yeast that play in roles of both Basidiomycota and Ascomycota. But as a human species, most familiar with the kind of the budding yeast, Saccharomyces cerevisiae, which a huge part of fermentation, leavening our bread, fermenting our beverages. Next slide. So kind of the very simple kind of budding yeast life cycle kind of is facilitated the fact that you can have yeast that can either go through the process of asexual reproduction or sexual reproduction. Obviously they're haploid, they have one set of genetic material, and they'll basically go through the process of a chemotaxis, trying to find a mate, basically kind of going through the process of going through the process of schmooing through this conjugation effect. However, if the yeast is unable to find the genetically compatible mates, it'll go through the process of budding, obviously creating a new formation of basically a new asexual formation. However, if you get through chemotaxis, if there is a compatible mate being found, go through the process of conjugation, kind of formally called schmooing, and kind of named after the comic since the formation looks very similar. And then you get the recombination through the conjugation to form a diploid, genetically distinct. This can go through the process of basically the spore formation, then going on the aspect of budding again. Next slide. So yeast are pretty interesting. They go through a number of different formation types. Obviously, when we talk about yeast, we're most familiar with the budding yeast. However, a big factor of whether yeast are forming our buds or pseudohyphae or hyphae are dependent upon a number of pressures by their environment, nutrient availability. And we most commonly associate or understand this budding yeast formation due to how we understand it. But there are multiple types of yeast that can form different morphologies and structures based upon their roles they play in the ecology. Usually, when we're kind of looking at Saccharomyces cerevisiae, we're familiar with the bud morphology, the pseudohyphae, and then also the colony formation that can occur. Next slide. So here we see an example of a yeast that can actually form a hyphal form. This is Yewaria lipolytica. This is actually a yeast that is pretty common in a number of soils, and so it shows that it can survive in the actual the budding form, the yeast form, but also these pseudohyphal, the hyphal filaments structures as well. Next slide.
All right. Now I'll talk a little bit about the saprobe life cycle, and just to say off the bat, these terms are not necessarily mutually exclusive, and those saprobes we're generally thinking of decomposer organisms, and then saprobe fungi, also called saprotrophs or saprophytes, but we're talking about things that eat dead material, and for the purposes of right now, we're really specifically talking about things that can survive off of dead material without a host. Because something about fungi is that they often don't delineate the ways we want them to, like to say, like, this is a decomposer, this is an endophyte. There's a lot of documented fungi that are sometimes an endophyte, sometimes a decomposer. And Dr. White was alluding to this a little bit last weekend with the rhizophagy cycle. And so we can have free-living organisms. But when we're talking about saprobes, we're talking about stuff that can survive on its own in the soil, decompose things without having some sort of host. And this includes things that we call molds, which are more or less filamentous fungi that grow mycelium that don't form mushrooms. And a lot of yeasts are actually saprobes too, because they will degrade things the same way that these filamentous fungi do, which we have a cartoonish schematic of their life cycle on the right there. But basically they produce spores. The spores germinate, hyphae grow, the hyphae fuse in the mycelium, and then if they're mushroom forming, they form a mushroom, or they just form more spores off their mycelium. And this is what it looks like up close, but we saw a lot of pictures of mycelium last week in terms of the microscopic stuff. But at this point in the talk, in case any of you watching have not already cultivated this skill, I want to talk a little bit about what I call fungal vision and being able to see fungi in the environment. And more than just the obvious ways, like you see a mushroom, that's pretty obvious. And so we'll start off by, looking at just raw mycelium. And so we see the white strands, and each individual strand we see is thousands and thousands of those hyphal filaments woven together. So here it's on the petri dish, it's on a piece of dirt. We also see mycelium like in our food scraps bucket if the right organisms are in it. Got all this white fuzz. That's some decomposer fungus too. All those little black pins are where the spores are producing on the hyphae. But there's also indirect ways to observe fungi. Well, we see these turkey tail mushrooms. That's pretty obvious. But also when you see wood that's bleached in this pale color, that's called white rot, because a type of organism called a white rot fungus has started breaking down the wood and the structure of it. So we see that means there's fungi there. Then we have brown rot, which is a secondary decomposition, which happens after white rot, where it's further broken down into the little brown cubey chunks that are like pretty brittle at that point. And when they end up breaking off and landing on the ground, they decompose and turn into soil. This is an old rotten tree that fell over, and we see at least 3 different types of fungi on it. We got these kind of grayish blue mold on the top, and then a few different colors of decomposer organisms. But basically what I'm trying to get at here is when you see wood or plant material and it's like turning colors like this and the structure is changing and falling apart, that's often an indication that you have saprobe fungi growing in it and other decomposers. And here's a nice, threads, more obvious threads of mycelium to look at. And to demonstrate the strength that this material can have, we have these 2 images where on the left, I'm holding up a pile of leaves, and the mushroom is dangling from the leaf pile because of the mycelial cords there. And then on the right, these are actually 2 pieces of wood. Picked up 1 piece of wood, and then there's another piece of wood that weighs at least a pound that's dangling from the other wood, and it's holding it there is a mycelium cord. And so if you picture having these in the soil, then you can start to see this is going to be good for the structure and stability. But at the end of the life cycle, a lot of these fungi, they grow mushrooms. Some mushrooms are pretty and iconic, and some are kind of a little more off-putting to look at, like these kind of flat mushrooms growing up this dead hemlock tree. This is the completion of the life cycle. And what's important to think about is that this tree is completely dead. It hasn't been alive for a while. And sometimes when we see dead trees like this, we shouldn't really view them as being trees anymore. We should really view them as being vessels for microorganisms. And then this tree is going to fall down onto the soil and go into the soil. So these are kind of the concepts of this like nutrient flow in and out of different media we want to keep in mind when trying to work with and leverage fungi in the landscape. And speaking of energy dynamics.
Yeah, and definitely that image that you saw last definitely showed an example of how some of those crust fungi can actually be the predominant recyclers in most floors. We usually kind of associate the fungi that produce macrostructures and mushrooms as kind of being there, but much of these unassuming or not so visually appealing are actually playing a huge role in cycling dynamics. But kind of talking about the energy dynamics as well, every single chemical reaction on this planet is powered by the sun. It's the fact that basically bacteria and algae evolved over time to basically harness the energy from the sun and perform the process of photosynthesis. Taking this radiant energy from the sun, photons, using those photons as an electron source, and then basically pulling carbon out of the air, being able to fix the carbon dioxide into various sugars of photosynthesis. So the whole process is pretty much this energy gradient. So plants, they are ancestors of these primitive photosynthesizers from algae or cyanobacteria, are basically going through the process of creating organic matter, through the production of their biomass, as we understand it, plants as their leaves, their shoots, their stems, the roots, but also their exudates as well. Whereas fungi are, played a role in understanding how you can recycle this organic matter. And so what our current understanding is that we understand about roughly about 4 to 500 million years ago, when plant life moved onto land, it was through an assistance of fungi, where you had these primitive protolichens, fungi providing the structure and durability of the thallus, and then providing a home for these cyanobacteria algae. There's a couple of theories about people how view this, but getting back to track, it's fungi play a huge role in recycling this organic matter, this plant material, whether it's at some stage of being viable, being moribund, or just, degraded, essential to recycling nutrients back to soil. Next slide. So we got a nice little example of a fungal ecology going on here. So we see various different types of fungi playing a role, but we see the aspect too is we see the autotrophic plants. Plants are autotrophs, auto meaning the ability to produce themselves. Trophic is, a fancy word of just saying how you acquire food. plants are able to photosynthesize. Like I said earlier, they're able to take sunlight energy and use that to drive the carbon fixation reaction that is photosynthesis. So naturally, through the aspect, we understand the roles of some of these symbiotic fungi, these mycorrhizal fungi. Obviously the arbuscular mycorrhizal fungi, they don't produce macro fruiting structures, the glomeromycota, but then you have the usually those glomeromycota, those are known as endomycorrhizal fungi inside the plant. But however, the ectomycorrhizal fungi, those are the fungi, the symbiotic mycorrhizal fungi that actually do produce macroscopic fruiting structures, and they form on the outside of the plant itself. Endo referring to internal, those are the arbuscular mycorrhizal fungi, or the glomeromycota, endo being internal, and then ectomycorrhizal fungi. Those are the ones that grow on the outside of the plant sheath in general. So you do get the symbiosis. Now, however, over time, through the production of this organic matter, whether it's actively through the process of leaves falling through the shedding of vegetation, you do have organisms that are basically taking advantage of this biomass that readily falls down, the kind of saprophytic litter fungi. These are the decomposer fungi that are actively recycling the nutrients that are being released on the forest floor. But then you also have situations where, with age as trees get older, they get the process of being moribund. You have the saprophytic wood fungi. These are kind of the primary decomposers that are breaking down the very tough cellulose or lignin bonds and forms and polymers together. So you get this whole process of ecological succession. So, even though the aspect is that through these continual growth cycles, it's essential that you have these fungi playing roles to decompose the freely available organic matter, but also decompose the organic matter that may take longer times or is more complex to break down. The understanding of the Carboniferous period, where the point where plants evolved the ability to produce lignin, where they were able to grow more than a meter tall, roughly a period of 300 million years, where you had these vast expanses of forest that would grow and the trees would fall down. And it's currently thought that microbes, bacteria, and fungi lack the enzymes to break down the complex lignin structures. We'll get into that a bit later, but it's essential that these different tiers of fungi, these primary decomposers, wood fungi, these secondary decomposed saprophytic fungi, and then also the tertiaries that are in the soil that do interact with with different types of symbiotic mycorrhizal fungi as well. Next slide. So kind of the fungal ecology, I kind of summated a bit earlier, kind of that kind of biochemical circle of life. But when we talk about this word biogeochemistry, it's the fact that when we are starting to study soil, a number of people are starting to realize how critical and important biology is. Usually soil was viewed by the extension of purely just geology, just by the structures that they were formed, and then also taking into account chemistry. But we're understanding that the biological organisms living on in and around that organic matter are also influencing these cycles. So the circle of life is there, even in both the biotic and the abiotic and the biotic, and they influence one another in dynamics, aka the circle of life in a nutshell. Next slide. So we're going to talk a little bit about kind of these enzyme degradation, decomposition systems. So how do these saprobes turn plants in soil? How do they basically recycle these materials? So we see a diagram here. This is a great little diagram, kind of illustrates a little more detail, different roles that fungi do play. So, breaking down organic matter that is otherwise somewhat recalcitrant, growing through it pervasively, but also freeing up these nutrients to provide them back to other plants that are alive. This is a huge dynamic of understanding that the network that is far more collaboration than competition, especially in dynamic roles between plants. When you have these advanced ecologies of further ecological succession, which is often predicated upon an advanced fungal to bacterial ratio. Next slide. So here we see a nice little example of, talking about when I'm using these words, lignins or cellulose or hemicellulose, kind of breaks it down. So, all modern plants, they have a combination of lignin, cellulose, and hemicellulose. What ultimately makes wood is lignin itself. It's just kind of this rigid, heterogeneous structure that does provide solid support. If you actually do look at the diagram, the lignin is kind of very much like these rebars, these structural supports. It's often interesting. You look at that molecular diagram of the lignin hemicellulose cellulose matrix. It often looks like kind of like a bit of this construction rebar, because this does provide a great amount of support. So whereas the cellulose is kind of, we're all wearing some form of cellulose, if we're cotton example. This is kind of one of the most abundant plant matters, but the reality is it's incredibly flexible. This lignin provides rigidity, but also too, it allows you to basically become a bit more recalcitrant. And we know this is very present among different types of plants. An example of whether that plant has a higher carbon to nitrogen ratio. Next slide.
All right, now we're gonna dive a little deeper into the biochemistry of how these white rot fungi are breaking stuff down, and not that you necessarily need to remember all this stuff, but just to kind of illustrate what's really going on at the tip of the mycelium when it's breaking down this plant material. And most of this is going to be based on research on the white rot fungi because their decomposition is pretty well studied. There's 3 enzymes that have been very well characterized that are listed there. And they all leverage reduction oxidation reactions, which are basically where they're passing electrons back and forth between molecules to stabilize or destabilize them in some way. Now on the right of the screen, see these chunks of white cotton candy looking tissue and then all these orange amber droplets. Those droplets are fungal exudates. So that's liquid that the mycelium is releasing to decompose things. There's other things in it too, and I'll get into that later. But the important thing to keep in mind is that the fungal stomach is outside of itself. It's like animals, we put stuff inside of ourselves to decompose it. Fungi just kind of release all their digestive enzymes and acids into the external environment, which has a lot of implications. But just give you a layout of like what one of these reactions might look like, because it can be misinterpreted when we hear about the fungi produce this enzyme and the enzyme breaks down this material, that you might actually picture the enzyme is like running into the bond and breaking it. But in reality, this occurs as a chain reaction where the fungus is oxidizing. It's producing this enzyme that gets oxidized, but then because the enzyme is too big to actually break the bond, it produces something called a co-substrate, which on the bottom right, that veracyl alcohol is an example of one of these compounds, that the enzyme then oxidizes the co-substrate, and then the co-substrate oxidizes the plant material. At that point, the plant material could break apart, the lignin bond or whatever, it could break apart, or it actually could combine into a bigger molecule. So this is not a very straightforward or linear process. There's a lot of moving parts, which is, it's really indicates why fungi are such incredible organisms. So here's a list of some of the co-substrates for these types of enzymes. And I circled the manganese peroxide because these co-substrates, they require manganese to function, or the enzyme requires a manganese co-substrate, meaning that if you don't have manganese in your soil, this type of reaction won't happen. So this is just a little glimpse of why having trace minerals and nutrients in the soil is really important and having them accessible, because in this situation, the fungus needs it to be able to decompose. And there's lots of specific enzymes that plants and fungi and bacteria use that require these trace minerals that they can survive without them, but they'll be reduced efficiency. And just one more note, it gets even more complicated with these ligninolytic, these saprobe decomposition reactions, because the enzyme they produce, like the lignin peroxidase or the lacase, is not stable above a pH of 4.5, but the chemical reaction to break down the lignin or to break down the plant material has to happen at a more alkaline pH. So the fungus, in addition to producing the enzyme and the co-substrate, is also producing organic acids to create a pH gradient in the soil from the tip of the mycelium to the thing it's trying to decompose, so that its enzyme can survive closer and then pass on its electron to something that can survive in an alkaline environment, or I should say not get degraded, because we're talking about chemicals here. Basically, the whole point of this is to demonstrate fungi are doing really complex biochemical things in your soil, and there's probably some level of biological intelligence that's challenging to just replicate through being a human putting in your own inputs. But to take it out of the abstract and into the practical for a second, this is what this saprobe decomposition looks like. And here's an example of it in practice. This is a bed that was made out of Japanese stilt grass, which is an invasive plant around here, and it was inoculated with the wine cap fungus. And then about 6 months later, this is what it looked like on the left. And that image of that brown stuff in my hand on the right is the only things that were there before was Japanese stilt grass and wine cap spawn. And so now it's turning into more of a topsoil material. So, if you have any sort of plant residues that might be viable for this, keep this kind of stuff in mind. And we're going to take a step forward from the decomposition and get into another really important thing the fungi do to survive, which is solubilizing minerals and nutrients from rock and soil surfaces. This falls into an academic field technically called geomycology, which stands for, like mycology, mycofungi, and then geo, like rock or inert material. And since we know that plants are not that great at accessing nutrients off of the surface of soil by themselves, they usually will employ other organisms to do it for them. How do these organisms, in this instance, saprophytic, do this for them? So here's another diagram, and we got the tip of the hyphae there, and then it's releasing an organic acid. Now, I mentioned organic acids before for creating this pH gradient for decomposition, but here the organic acid is providing a different function. It's being used to dissolve minerals off of soil and rock. So basically an acid is a molecule that has a hydrogen atom or a proton that can easily dissociate or leave the molecule. So it's reactive. So when that protein leaves, when we see on the diagram, it will then go to the surface of the soil, take the place of another ion that's already there, and then make that soluble and then available for other things to consume. And so in the diagram, we have calcium, potassium, manganese, some of these things there. The other side of it is when the acid dissociates, the anion, the part that lost to the proton, will then probably bind to a different ion in a process called chelation. And for example, it could be manganese. And so this acid could turn into one of those manganese chelates that is then necessary for decomposition. So there's all these kind of feedback loops that are happening and co-occurring with the fungi. And so, a good example of geomycology we can see, which is the beginning of the ecological succession, is when you see lichens growing on rocks or human statues, because the lichens contain fungi that have these strong acids, and then will eventually turn them into organic matter in soil. And probably the most commonly observed and well studied of these organic acids is oxalic acid, which you all probably have heard of in other contexts because it's a really common throughout the biological world. And just to give you a schematic of this kind of acid dissociation, the oxalic acid, it loses its protons. Now it's got these open charged groups, which then will bind and associate to some other ion, often a mineral or a nutrient or a metal. And so we see the little stick diagrams, but what does this look like in reality? These are some images taken from cultures of fungi who were provided different nutrients, and we can see that like the calcium oxalate turns into these distinctive double tetrahedral crystals, and then these other, the copper and the zinc, they form these kind of distinctive looking, what we call precipitates when they form. And here's some nice images of these, metal precipitate crystals growing directly on mycelium. And so we're seeing this process happening right at the surface of it. So it's these processes of mobilizing nutrients and decomposing things, which really highlight how fungi survive and also a lot of their importance to the landscape. And now we're going to get into how these are important for the health of plants and the ecosystem at large. So nutrient cycling, I've just been going on and on about that for a while, but just to recap, the decomposition can release carbon-based nutrients, but then also the geomycology, the acid dissolution will make other macronutrients, micronutrients available. And so that discussion's really been about like the filamentous fungi, the mycelium ones. But yeast actually are really involved in this too. Yeast do a lot of this nutrient cycling, but they're not very well studied. There's a pretty, there's not a whole lot of scientific literature on the roles of yeast and like soil nutrient cycling, but studies that are out there demonstrate that they are doing it. And so there's evidence that they are solubilizing, making available things like phosphorus, calcium, and zinc. Some yeasts actually produce ammonia on their own, which is basically would allow them to be fertilizing plant roots directly. They also produce things called siderophores, which will chelate iron and are related to the availability of iron as a nutrient for plants and other organisms. And then they also create degradation enzymes too. So the yeast can also be saprobes. In addition to the nutrient cycling, soil yeasts also have been observed to produce plant growth hormones like IAA and IPYA cytokinins, and studies have demonstrated that certain types of soil yeast, when they are there with the mycorrhizal fungi, the mycorrhizal will colonize plant roots better. So there's like another one of these, kind of multiple feedback dynamics, complex ecosystem interactions where it's not just do you have good mycorrhizae, what's also in there, because some of these other yeast and organisms can influence that. Now, another benefit for plants, besides the nutrient cycling, is to support the plant immune system and prevent pathogens from growing on them. And they can do this in a variety of ways. One is through the production of biofilms, and this is some images of it's an apple that got a wound on its surface. And then this yeast, Saccharomyces cerevisiae, that same brewer's yeast species, grows into a thick biofilm over the wound and then makes it so that a pathogenic mold cannot grow into it. And so yeast biofilms are important for protecting plants in this way, and they're also important for plants or soil in other ways too. Biofilms are pretty important things. But in addition to the biofilms, yeast can also produce antifungal compounds. They can use their siderophores to starve pathogens of iron by basically taking all the iron so that the pathogenic microbes can't use it. They can also directly inhibit the growth of them. They can break down the proteins and the cell walls of pathogens. They can directly attack them. And they also can cause yeast and mycorrhizae and other types of fungi will actually tell plants to regulate their immune system better than they would on their own, and they'll actually do things and influence the release of chemicals like salicylic acid and jasmonic acid that are important for plant immune responses. In addition to all those kind of nice little pat on the back helping out the plant things that yeast do, and also that the filamentous fungi and the saprobes do, there's also predator fungi that will directly attack and kill and eat soil pathogens. And so we saw this picture last weekend that Chris Trump showed us from his microscope session. And it's one of these noose fungi that they have these little rings on them, which are made of 3 cells that when a nematode goes into it, they swell up with water and contract and then strangle it and eat it. But in addition to some of those interesting, more ornate looking fungal predation, a lot of the common fungi we would work with as mushroom cultivators also can eat nematodes as well, including this one, the wine cap, which I've already mentioned before. It produces these things called acanthocytes. They're little spiky balls that it'll produce, drop into the soil, and then they'll puncture the cuticles of nematodes, and then the fungus will eat their insides. So there's a close-up of them getting poked and bleeding out. And then in real living color, here's what they look like. We can see the white little spiky balls. And then on the right, those little clear tubes are nematodes that got stuck to them. And now they're probably going to become fungus food. And now we're going to get into soil building a little bit. Another function.
So here's a nice little diagram. It's from an environmental engineering firm. Although, obviously, building of soil isn't of actual construction, so that was an interesting little image to play off it, but we'll kind of get into details. Next slide. So we understand that the critical aspect of what makes soil, besides biology and organic matter, is the structure. And this is the factor that, having structure, having these passageways for air infiltration, allowing water to pass through it, but also not run straight through it is essential. And it's facilitated by the interactions of, one, organic matter, your mineral soil particles, and the factor that you're getting the microbiology, mostly bacteria, to make these alkaline glues that will make these microaggregates, and then you're getting the fungi to combine all these microaggregates together to make macroaggregates. Kind of a good analogy to think about is imagine the Brooklyn Bridge. There was a bridge built in the 19th century, made of these huge stones. These were all kind of built by individual humans. They had these huge wrenches that were 5 feet in height to build these microaggregates, but you have these giant steel girders holding them all together, and it's the stressing effect, the tension that holds it up. So the fungi are playing a huge role to basically build the structure in the soil. So the biology is critically important, but not only just the biology, but also the presence of fungi being the architects of the soil. Next slide. So we'll kind of build some perspective when we talk about what fungi are, filamentous fungi in this example. So we see that fungi are pretty interesting because they are inside this grand network of these filamentous structures where you have a cytoplasm where all these different organelles are living inside. But if you actually want to cut away down to the actual structure, the majority of fungal biomass is mostly carbon. So the outer pellicle of the mycelium itself, these hyphal tips of these manoproteins, these are pretty much glycoproteins. So these are pretty much mannitose bound to a protein itself. Then you have these alpha and beta glucans. So these are incredibly large molecular weight sugars that pretty much make these cross-linked hashes between the manoproteins and the chitins. To give some reference about how big these sugars are, when we talk about like a sucrose molecule, which is a glucose bound to a fructose that has an atomic weight of roughly 200 to 300 atomic masses or daltons. When we're talking about a glucan, alpha beta glucan, these are in the order of 2 to 3 million atomic masses. These are huge, they're enormous. You're talking about maybe a sugar molecule, a single about a person. A glucan is like looking at a small city. Then we have chitin, acetylglucosamine. This is the polymer that fungi are made of. Crustaceans also have them. Crustaceans, however, biomineralize. But this is basically what makes fungi tough and somewhat heat resistant as well. Also the reason why you have to cook all your mushrooms that you do eat. We don't have chitinases in our stomach, so heat helps to break them down and also to expose, obviously, the cell membrane, the phospholipid membrane, which we have the hydrophilic phosphorus tails, and then the heads, and then we have the hydrophobic lipids on the inside. So we understand the cell structure that fungi are interesting, but also the vast majority of their biomass is made up of a huge carbon. And as they grow further out, they're going to be continue to build the more distant parts of the mycelial network with more mannins, with more chitins, and continue to reinforce. So as the distal tip of the growing hyphal filament will have a much narrower carbon to nitrogen ratio, the older parts will have a much wider, way more carbonase to reinforce these cell walls. So the aspect too is, so when we talk about carbon sequestration, fungi are critical for this because they're able to take the organic matter and basically lock it in place. Next slide. And then also talking about this as well, when we talk about, carbon, soil carbon, there's all these different notions. And it's really important as we're kind of talking about how do we actually draw this down from the environment. As much people thinking of it as an engineering solution, biology has been doing this for hundreds of millions of years. So when we talk about what is humus, right? Humus is the organic kind of layer of the soil. So what is it made of, right? So when we talk about a humic acid, it is kind of a large structural heterogeneous macromolecule. So if I'm going to show you like this, a picture of humic acid, well, this is kind of like an idea of what it looked like. A kind of humic acid is this amalgam of all these different complex structures that, are that basically fungi really only have the enzymes to break down. These are complex organic materials. This is why fungi are so essential for accessing these. So you could have really good organic matter in your soil, but without the biology, those nutrients are going to be locked away. When we talk about the ability to more so lock away or mobilize certain types of high concentrations of heavy metals, humates are essential. This is also known for actually building compost. If we have a water supply that is high in chlorine and chloramine or other kind of heavy metals, a really great way to clean it up is through basically you adding humic acid or treating it with a compost that has a sufficient biomass matter as well. So when we talk about the humification, it's the continual cycling and digestion and exudation and digestion and exudation of this organic matter, basically by microorganisms, but largely facilitated by fungi. So humification of the organic matter. Next slide. So kind of talking about like practical soil building, right? We talked a lot about a theory, but we're going to give you a little tease about how do you actually build soil. We'll cover it more on the actual practical applied aspect later in the presentation. But this is a project I was involved with back in 2019. This was MS-126. MS-126 is the Magnet School for Environmental Engineering, Greenpoint in New York. This was a program called Eco Schools. This was set up. Next slide. This was set up as an initiative to the Greenpoint Community Environmental Fund. Greenpoint is a neighborhood in New York City. It's right along the Newtown Creek, which is estuary off the East River that basically separates Queens and Brooklyn. However, it's also been the site of many oil refineries from the greater part of the 19th century. So basically you have on the bottom in Newtown Creek, you have a layer of organic pollutants called black mayonnaise, which is 6 to 7 inches thick. So in general, BP went, basically these refineries were once owned by Standard Oil, and obviously Standard Oil got broken up. These assets basically got purchased by BP, and there was a settlement to basically force some accountability for this. Anyway, but I was working with Faye Walker, who is the sustainability coach MS 126 under the National Wildlife Federation's Eagle School program, to basically talk about the role of biology in the soil. They had a composting setup. They had an outdoor experimental permaculture garden, but they didn't really have that much connection about the roles that fungi, that biology are playing in the soil to build and the soil cycle itself. Next slide. So here's an example of what some of that soil, or more likely dirt, from Manhattan Avenue Park right along the Newtown Creek. We see lots of compaction. we see no fungi. And we'll show you a little bit video of this a little bit later on, where it's literally pulsating. It's so concentrated with bacteria. Next slide. So this aspect too is to basically work with the students to kind of teach them about not only identifying how important soil is, but also how they can understand the biology that's living in it, but also hands-on experience of building soil. So they had a pretty solid thermal composting system. Pretty much it was a system that was solar powered that would burst air through the compost pile, so it was thermophilic. However, this possibly maybe oxidized a bit of the compost too much. One aspect with compost is that oftentimes people don't let it sit, mature, and cure, where when you get it to thermophilic, it's really essential to killing all the potential pathogens, pests, and also different seeds of certain plants you don't want in your organic material. But if you don't let it cool down and mature, the populations of beneficial aerobes aren't going to recover. So this was an example where we pretty much taught the students about the role that fungi play in the soil, but also understanding that saprobes are a critical component of this. So we had this with some photos from our demo day. So Smallhold is a mushroom farm in New York City. I have good relationships to us. They donated to us roughly 150 pounds of mushroom blocks that we went through the process of when they were basically prepping the raised beds for the start of the season. We scraped off the top layer of soil from the raised bed. We laid down compost from the thermal composter. We laid down mycelium from the spent mushroom blocks, sawdust, cardboard, and pretty much did this lasagna build up. And then we covered everything with mulch. So we basically, to make sure it basically was covered, and we watered them. Next slide. So over the course of the month, basically the mycelium from these white rot fungi, these oyster mushroom mycelium, grew together, grew through the compost, grew through the cardboard, the sawdust, the residual, nutrient material that was left over from the actual blocks themselves, and weaved together and basically fruited over the course of the month. We basically did that install in April, and then basically at the start of May, they fruited these huge oyster mushrooms on top of the actual raised beds that we set up. So we had pretty much this aspect where on top of the mulch were these huge mushrooms that you could basically this anywhere from the size of a dinner plate to basically you could hold them in your arms and your hands, and this was amazing. And if anyone is a fan of culinary mushrooms like cooking, maybe we want to be tempted to go ahead and pluck some of these. However, we just let them just sit in the soil. Next slide. So some people may think, oh, what a bummer mess. Well, the thing about it, maybe we can do an aspect of using these decomposer fungi to, help assist in the restoration of the beneficial organisms that are maybe gone dormant in the thermophilic compost. Because the reality is we let it sit over the course of this month, added in a bunch of fungal foods, inoculated with a bunch of beneficial, mycelium from oyster mushrooms, which are white rot. So we basically allowed them to mesh together. And what's even better is after they fruited, they sporulated, but also too, we didn't pluck them. We let them sit. Basically over time, they started to decompose. That attracted insects. The insects basically feasted on the mushrooms and reproduced on them. They laid eggs. Eggs hatched. The eggs turned into pupa and larvae that attracted birds. And you're basically getting the restart of basically a fungal ecology by simulating the succession of basically this decomposition cycle in this raised bed. Next slide. And so even further, basically, we go through the process where we basically taught the students to understand how this process actually functions and works, so they are able to understand the role of soil biology. Next slide. Julie, I do talk super fast, so I'll try to slow down a little bit. I tend to enunciate rather quickly. Anyway, but yeah, that last image was the example of the large mushroom. But here we see a sample of the actual raised bed after we went through the process of basically the mushrooms sprouting and growing off them. We see lots of great structure. We see a plant root. Next slide. We see some hyphae coming out from the plant, basically from the plant material that was left over from the compost. We see lots of good structure, airways, passageways. Next slide. And then we even see some microaggregates. We can see a couple bacteria in there. We can even see that these are beneficial fungi because they have that clamp connection. That's a little nodule, that little bump. So it's really amazing that we can really work with fungi, these decomposer fungi, to basically boost the quality of our compost, or basically take beds and to kind of build this kind of living soil by emulating what's happening in these advanced, these successional ecosystems where fungi play a huge role. Next slide. So we're kind of going over a bit of the ecological succession in soil. We'll kind of go over the soil food web, some plant soil succession sequence, understanding what types of plants are, how they thrive at different points of succession, and also some of the fungal indicators of soil succession. Here's a nice little diagram kind of talking about this primary succession. Soil food overview, kind of demonstrating the different areas, environments that there are within these little microcosms of soil that do exist between this biomass. Next slide. So this is a slide from the NRCS soil primer, kind of definitely illustrating the understanding of the interactions between organisms in the soil at the basically the different kind of tiers. But these interactions are, there's even further interactions that do exist between them. This was more so the image that was put out at the time when the primer was published. But we understand the basic soil food web explanation as being the dynamic that you have the sun providing all of the energy basically through this entire gradient, and then biology basically allowing to take different steps of this gradient of energy down the chain further as well. So obviously plants being the first trophic level, being able to basically fix all this organic matter, and then we get to success levels. So when we talk about the soil food web, to our left is kind of more an example of what soil looks like. This was a little better diagram because it actually gets into some of the horizons, and the plants actually do get down into the subsoil. A lot of notions of plants, say, oh yeah, they're just kind of living on the O horizon at the top of the A horizon. But on the right, we see an example of what soil is. It is a robust, living, and dynamic ecology and ecosystem full of life. And so we need to understand that example. So this is kind of a macroscopic view of the soil, but if we zoom in the next slide, we see the microscopic view of the soil. So we understand here that when we see compaction and everything is smashed together, there's no room for life. There's no room for passageways. There's no way for water to get through, or if it's, if we're working with a much more of a clay soil, if it's much more of a sandy soil, it'll just pass right through. But is the essential role of biology in the interaction with the organic matter and the inorganic matter to build this structure. So we're starting to see an ecosystem, a neighborhood over here. It's much more dynamic. And if I kind of think about if you have take an aerial kind of snapshot of an old town from a city where people have been living for hundreds, if not thousands of years, you see all these different kind of unique winds and weaves of organic structure. Whereas kind of maybe a lot more of the dirt looks kind of like a Leviton, kind of a cookie cutter cul-de-sac place where there's not much flow through. So we can kind of imagine it's better to have this natural organic process by which biology is able to interact with it as well. But pretty kind of good overview snapshot of it as well. So next slide. So going back to the point of the slide, kind of talking about how we see the structure in the soil much more easier on the eye. You can see the dynamic roles. You see organic matter. You can see microaggregates. This one's a little bit out of focus, but you get the idea. But we have a video to kind of show you a further example. So this is a little video of some microscopy. So we see little nematodes zooming along. We see all these nice little structures. We see sand grains, microaggregates, macroaggregates. He's going to play it a few more times, but we see an actual dynamic ecosystem that is going on. And this is essential when we see in soil, because with this compaction, you can't have anything going on. It's static. Really, it's going to go anaerobic, and the only thing that are going to thrive there are bacteria that are going to be able to survive on these lower oxygen gradients. Next slide. We get a little more zoomed in slide, a little more nematode action as well. Dynamic structure, we see bacteria, we see roots, we see mycelium, we see nematodes. And so this kind of give an example if you haven't looked at underneath. Oh yeah, and then here's the exact contrast. An example of dirt compacted. There are maybe little bits of organic material, but there's not the biology which essentially break it down. And then we see here, if you pay attention, you'll see the pulsating of an incredibly concentrated basically slide preparation where literally you can see the bobbling Brownian motion of the bacteria interlaced. So we understand that, that this is dirt. There is life in it, but it is abundantly bacteria, and there isn't the essential biology necessary to facilitate this nutrient cycling to exist. Next slide. Cool. So we're going to talk a little bit about, in the soil food web, we understand that there are these different interactions between the trophic levels. However, there are also intra-organism interactions. So something that both Leif and I are definitely interested in are kind of the interactions with different organisms within, especially with fungi, since we do have that kind of fungal background and perspective. So definitely this next little section is going to focus on the interactions between bacteria and fungi. So when we talk about bacteria and fungi, that there's a number of different roles, that there's, basically bacteria can facilitate in sporulation release, growth, hyphal development, protection, antibiotic resistance, and nutrient acquisition. And basically it talks about bacterial fungal interactions. So this is a nice little overview slide from a study that we'll kind of list in our references later on if you want to dive into that. Next slide. And we're going to play a quick little video. This kind of talks about some really interesting interactions between bacteria and fungi. This is Pseudomonas pudia, basically these little bacteria moving along this little water filament on a fungal hyphae. So it's amazing because these fungi are acting like superhighways by allowing the bacteria to get around. So almost like the expressway. And kind of when you think about soil, I kind of imagine this outer space environment. There's asteroids and meteors and just floating matter in the soil kind of continuum, or basic biology has to kind of move between these little inorganic materials, but also on other organic materials and organisms to get through. So it's pretty amazing because in general, fungi inside the hyphal filament, you have the cytoplasm where they're moving their own materials, but also their own information through basically multiple basically in the fact they're multinucleate, cycling all their different organelles, but also you have the Pseudomonas pudia, which is these little bacteria which move along the actual films of water that's suspended on there as well. Next slide. So also too as well from this study as well, we talk about an example where they tagged both bacteria and fungi with a fluorescent dye. The fungi is tagged with a green fluorescent dye and the bacteria are tagged with a red fluorescent dye. So you can see that bacteria are literally cascading over basically in the wake of the growth of the fungi. So this is more so kind of showing the filter was showing more of the red, but you see the bacteria are kind of coming in a wave along them, and basically playing a huge role in facilitating the external metabolism. Fungi are producing these enzymes, which are kind of floating out there in general, which are able to digest a complex number of food sources, whereas bacteria usually they're good at breaking down a wide variety of things, but can only focus on one thing at a time. So you get this co-metabolism going on. Next slide. All right. Also, as well, another example of some fluorescent microscopy where you see waves of bacteria kind of going along in the wake of the fungal mycelium as well. And it's pretty essential because we understand that there's a pretty huge role of the bacterial fungal interactions in the soil. Next slide. Yeah, so this kind of is a great paper which does go over some of these really fascinating aspects that, even sometimes too, the bacteria can play a huge role in facilitating the growth of basically the fungi. So the distal tip of the hyphae, is called the Spitzenkorper. It's a really unique one, amazing word to say, but also it's a really unique aspect where the distal tip of the hyphae is being broken down and reformed as you go. And what's even more interesting is you basically have, on this study, they showed that the bacteria will actually go through the process of its phloem, will move along the hyphal filament, and then supply the fungus with thiamine. I believe thiamine is one of the vitamins in the B complex. But also, too, what's even more amazing is that there are even some thiamine riboswitches. So you have some elements within genomes that can be stimulated by transcription factors based upon certain elements. So riboswitches are RNA molecules that basically can change their structure and conformation and then alter different gene expressions. So this is kind of a really interesting kind of modification of gene expression, which is huge, especially at the distal tip of the fungus, which it's going to be choosing which ways to move in direct. And then even further, the fungus grows, continues to obtain nutrients and environmental niche. And this is kind of this nice little symbiosis. I kind of think about like a little pilot fish moving along a shark or a whale, how they're kind of living together. But we see the kind of loop between spatial interaction, metabolic interaction, this bacterial fungal mutualistic strategy. Next slide. So yeah, even to some more interesting aspects as well, the yeasts, the fact that certain microbes do play, that you have other interactions between fungi, bacteria, and protozoa that are being facilitated interestingly as well. Next slide. So even too, we do have different roles of yeast biofilms. Definitely in understanding playing a role in protozoa food. Even though cilia do show up in higher anaerobic environments, we understand that going through the aspect where the soil is going through a process of a compacted lower oxygen gradient to a higher oxygen gradient, that these yeast can often be a factor of these biofilms being a pathfinder species, digesting organic material while freeing it up for the aerobic microorganisms. So pretty much you have the aspect of this yeast Cryptococcus laurentii biofilms kind of being viewed through basically standing in fluorescence, and then show that these different fluorescents exhibited on the inside of ciliates as they're being digested. So the consumption of these biofilm is being consumed and moving along, and that just kind of talking about, we're only kind of scratching the surface as we're filling out the the inner kind of lacing, meshing of the new interactions between organisms and the soil food web. So definitely, this is kind of, as we're starting to understand the roles that the yeast do play in soil between nutrient roles, exchange, and facilitation. So we're only just at the tip of the iceberg.
Okay, a little interlude here. Efren talked about this during his talk, how there's not a whole lot of academic research on the soil microbes as they relate to the cannabis plant for various reasons, but there's starting to be some research out. We couldn't really find too much about the soil fungi and how it relates to this specific type of plant, but there was a paper published last year where they were surveying the fungal communities amongst 3 different cultivars, couple CBD cultivars and a high THC strain. But what they found, they couldn't determine too much because it's a pretty complex field to research. But what they did show is, if you look in the upper left-hand corner, that the highest diversity of soil fungi is in the rhizosphere. It's in the soil around the roots, more so than it is inside of the roots or in the stems or the leaves, which could indicate that the soil is a reservoir of microbes that the plant might use. And then that might tie into kind of what Dr. White was talking about last week with the rhizophagy cycle and how the plants are selectively uptaking certain microbes into them, either to harvest nutrients or maybe turn them into endophytes for other benefits. And on the right, this diagram is listing some of the endophytes that have been identified in various parts of the cannabis plant. The blue are bacteria, the red are fungi. And what's interesting is that some of the fungi listed here, like they have Schizophilum, which is a white rot fungus. It's a wood decomposer. It's never been observed as an endophyte, but it lives in the plant. Maybe it's an endophyte, maybe it's just waiting to decompose it later on. But also a lot of these molds, like a lot of the Aspergillus molds and things like that, could potentially exist as a soil mold as well as inside of the plant. So in terms of, we just want to think about like the plant and the soil being very interconnected and influencing each other's microbiomes. And the authors of this research even speculate that even from doing a clonal cutting from a mother plant is still going to exert a profound influence on the soil you put it into. So it's like, once you get the good soil biology and the internal biology, they can keep kind of inoculating each other.
Yeah, and definitely we'll get into the aspect of, the plant soil succession. So kind of the example of rock to forest, kind of on that similar note of the kind of transmission that Leif mentioned. We're realizing that this vertical transmission of endophytes from progenitor to offspring, or from progenitor to progeny, from parent to offspring, is pretty essential. And also, too, to think about, that these lichens, when we talk about them being the active ones at the start of an ecosystem, that they're basically even some thoughts that they're kind of little ecosystems in a thallus, that they're even kind of the reservoirs of biodiversity to help kind of spread this, biology and nutrient cycle even further. But that's for another talk, different black hole. But yeah, getting into the overall understanding about how we get from this process, we get the initial wettering condition where there's erosion that plays a huge role, but also, this primary erosion that does occur basically does facilitate the colonization of these rocks by algae and lichens and fungi that form these lichens being the association between a couple different types of fungi and algae or cyanobacteria. But it's essential because you're getting the first generation of the organic matter, the exudates being produced by these algae or cyanobacteria, which helps build the organic matter of the soil over time. You're getting this gradual humification. And then even further, you're getting the advance of more advanced plants through time. You're getting more complex exudates. You're getting plants that tend to be putting more nutrients into their roots. They're going from the process of being annuals to perennials. you're shifting from plants that are annuals, which put more energy into vegetation and seed production. You're getting perennials where the plant is going to be putting some investment back into its root system year after year. Then you get further basically breakdown of organic material. You get the process of humification over time, and then you get the building of topsoil. However, through human interaction over time, often kind of starting the lower land estuaries, you're getting the growing on the kind of the rich sediment off the water systems, and you get this terracing effect as you move the hill, and tend to get this erosion that's pretty prevalent from across the world. But that's an interaction between geological, to ecological, to anthropological timescales. Next slide. So here's a nice little diagram. It goes over the kind of the nice aspect of progression. I like this diagram because it kind of shows you the kind of through these kind of sinusoidal examples of how important these different focuses are. You get into different types of biology, different types of roles. We have bare rock to mosses and grasses and perennials, to woody pioneers, fast growing trees, and climax forest. And that's kind of the indicative, like we showed you previous, was the primary succession. However, through a disturbance event, whenever there's a fire or earthquake, let's say kind of like a construction or building, that's secondary succession. That's kind of where there's been a disturbance that happens through naturally or artificially, and then biology is going to basically recover and going through the whole successional process again. Next slide. But kind of talking about these indicators of soil succession, it's understanding the factor that, you're getting obviously these kind of these fungal to bacteria ratios. I know someone in the chat was kind of curious about what happens fungal to bacteria ratio. When we're talking about these fungal bacteria ratios, usually we're talking about filamentous fungi. These are the macro fungi. When we see them on microscope slide, they're substantially larger than bacteria. Yeast do play a role, but we just haven't characterized enough that role to that date, but we understand they're pretty important for kind of priming that. So kind of yeast are playing a function in that kind of early successional stage pretty critically as well. But yeah, kind of going through the process, kind of going through the primary succession, we get these pioneer species. We're getting lichens, which are these aggregates between bacteria and fungi and cyanobacteria. We're getting the annual plants. We're kind of seeing fungal to bacteria ratios of 0.1 to 1, kind of the weeds. Predominantly the nitrogen cycle is going to be nitrate because it's going to be more alkaline, bacteria producing more alkaline glues and that capacity in general. And then that's kind of keeping the mineralization to that point where the nitrogen cycle is going to be more nitrate versus ammonia. Then we get further on to the grasses. We're getting more of a fungal to bacteria ratio of 0.3 to 1. To that point, we're seeing far more grasses. We're starting to see more kind of to the point where we're starting to get further along in the succession. We're starting to get more fungal biomass. So the mineralization factor is going to be more shifted where you're getting both nitrate production and ammonium production by basically more of an acidic profile by higher presence of fungi. Then you're getting further into shrubs, 2 to 1 to 5 to 1. You're getting more the first kind of stages of perennials that are present as well. And then further getting to further succession as well. So we're getting to that 5 to 1 to 100 to 1 ratio where it's predominantly fungi, which are producing far more acidic exudates in that capacity. And that's kind of shifting the mineralization where it's going to be mostly ammonium at that point. Next slide. So kind of going to the example, the next aspect 2, kind of to recap again, because iterative schema, process learning, we get the dirt to soil, and we're seeing structure to that capacity. But understanding that role that fungi play a huge role in being the architects of the soil, that fungal bacteria ratio is essential, especially when you need to, especially when it comes to further succession. That this is going to impact the nitrogen cycle in phase as well.
All right, well, we made it through the theory. Thanks for bearing with us through that. But now that we've gotten a little laid the groundwork of what fungi are doing and why they're important, we'll talk about some. Most of them are pretty straightforward things you can do to work with fungi in your soil. With the preface of this whole section is that, kind of leveraging soil fungi is still a relatively unknown topic that, there's a lot of mysteries still to resolve with it. But these will be some guiding points you could incorporate if you want. So start out by what I call saprobe soil inoculation. And this is what I'm referencing here is where we're working with one specific type of a decomposer fungus and then mixing that into our soil. So usually you would start with some sort of what we call mushroom spawn. And on the right, we've got some of these 5-pound spawn blocks that mushroom farmers produce, and they would, either cut them open to grow mushrooms out of the side or break them up and mix them into another substrate. So for most saprobes, and a lot of this discussion is very biased towards white rot fungi because most of the gourmet edible cultivars are white rot fungi, and they're the ones that were primarily have been researched for degradation and bioremediation and things like that. But generally, kind of food that works for them is woody stuff that's not so good for bacteria to eat. So we're thinking, like sawdust, wood chips, straw, even paper products, cardboard. And there's various types of these to use. But probably the most vigorous ones that are easy to buy commercially are the wine cap, which we already mentioned, and then the oyster mushroom, which we were seeing a bit in that soil building demonstration that Craig was showing earlier. So these two, in addition to being tasty, edible mushrooms, also do a lot of decomposition. They both eat and consume nematodes in the soil. They both can live in the soil. Even though the oyster fungus, we usually think of it as a wood decomposer and growing off of trees, it can have a fully functional life just in the soil as well.
Leif, do you want to just quickly explain why we call them white rot or brown rot? Because I think we mentioned it earlier, but we didn't talk about what is being decomposed primarily or secondarily.
Yeah, no, we went over that at the beginning, but just to remind, the white rot, we showed it earlier where the wood looks kind of bleached. Again, that's the primary decomposition because these fungi are breaking down lignin, and they're the only organisms we're aware of that can really effectively and efficiently break it down. And then there's brown rots that are secondary decomposers. And then some of the edible cultivars are brown rots, but not as many of them. But just to give you a quick, just practical how you could do this and mix this into your soil, you can just take a spawn block and break it up and mix it into your soil media. If, say, you're growing plants in pots, adding a little bit of sawdust or something can help stimulate the fungus, but it's not necessarily needed because we're not doing this to grow mushrooms. We're doing this to get more fungal and biological activity in the soil. But here's just an example of, this is an old tobacco pasture where we're trying to grow some perennial herbs, and it's all packed, compressed clay soil with poor nutrients and all that. So throw in the amendments and add some core, some compost, some sea mineral fertilizer, and then just broke up the Stropharia spawn and mixed it in there too. A few months later, we can see this in the bottom right. That's the soil profile, and you can see the Stropharia mycelium growing into it. So it's already starting to get active and do its thing in there. And then next season, we can go and inoculate it again, and then cover it with some leaves. If you're adding fungi to your soil, you always want it covered. You never want your mycelium to be exposed to the air or sun because it's going to dry out and not grow. I'll talk a little bit more about that later. But also throughout this process of inoculating the bed, and I was putting the sheet mulch around the edge. Now the sheet mulch is all covered in mycelium, so it is becoming an inoculum. So then we actually can take that cardboard and use it to inoculate soil further and bury it. And then we got the mycelium growing in the soil. We got some nice cords there. Got this big mama wolf spider moving in on the plot. Like kind of field of dreams theory, building there will come. Maybe think integrated pest management, because this is a voracious predator that loves eating small bugs, and it's got hundreds of little babies on its back that are going to do the same thing. So we're thinking about ecological farming here. And then, we get some nice growth of, this is a perennial medicinal herb, not the one you all are probably as concerned about, but that's the gist of it. But you might run into the issue, though, of, you want to inoculate your soil, but it costs $20 for one of these spawn blocks, and you don't really want to buy a bunch of those blocks. It's a pretty big expenditure. But the good news is if you're working with these species, and particularly if you're working with the wine cap Stropharia, it's very easy to expand this yourself, as long as you get some hardwood chips of a wood that doesn't have a lot of antifungal compounds in it. All you have to do is soak some wood chips in a bucket overnight, layer them, drill some holes in a 5-gallon bucket, or use a tote bin or whatever containers you have, and then just get your spawn bag, layer the wood chips, the spawn, and then a few weeks later, it'll probably look like this. And now you've gone from having, 1 gallon of spawn to 4 gallons of spawn. And then you can just repeat this process over and over as long as it doesn't get contaminated. So that way, if you want to start, and this is something you can mix with mulch, you can mix into your soil. So this is it can be a helpful tool for your increasing biological activity in your soils. And you can do this with other things too, like paper. Paper and cardboard's not not as ideal to put into your soil, but same concept. You soak it, drain it, inoculate it, and then now it's covered in mycelium. And then you can use it as a vector or to, throw in your compost, on your soil and whatnot. So, yeah, next we'll talk about spent mushroom substrate. We're going a step deeper in now.
Mm-hmm. Definitely. So, as is occurring right now, there is a pretty large expansion of gourmet mushroom cultivation going on. Formerly most people, the further experience of a gourmet mushroom was maybe a button mushroom on your pizza or on a salad. However, this has kind of come about through kind of the DIY cultivation that has occurred probably since the beginning of the 2010s. But yeah, so it's great because there's a wide variety of mushroom cultivators that, this is often kind of a burdensome waste product where there is there's basically a large amount of just available kind of on hand. So oftentimes there are a number of people are concerned of like, oh, we're going to buy this spawn. It's going to be expensive. Oftentimes this is an abundant resource that mushroom cultivators are doing. Now, if you're thinking about sensitivity for what's in this material, it's always good to follow up with the mushroom provider, kind of talk to them where they source their blocks, they make them in-house, but kind of talk about the reality in general that, to think about this for use, it's pre-digested, biologically active ground. They are considered kind of more of a green since there is a fair amount of nitrogen still living in the biomass, but basically it can be treated also too as a vermicompost ingredient and also a substrate inoculant. You can use this to inoculate your browns, which will facilitate the biodiversity prior to composting and even further during the maturation state, but basically during the maturation phase after the temperature is stabilized if you have working compost. A note on compost as well, certain recs for when it comes to compost are going to be a little more strict about what you're adding to it after it. But if you're basically working more in just the line of your soil in that capacity, this is a great addition into there as well if you're using in certain capacities. Next slide. So in general, oftentimes if you're going to go visit a mushroom cultivation operation around your area, you'll often see this large pile of mushroom blocks put out back. Oftentimes, a number of people that are producing these, they'll flush the mushrooms off once, and just kind of from a commercial standpoint, they're often growing these in a controlled environment with set humidity and temperature conditions. So the notion of once they cut that bag open, it's no longer hermetically sealed. So the factor in general is they're kind of flushing it once, and oftentimes the organism will respond. Also, you'll get secondary flushes in the block itself. But it's kind of an amazing aspect that you have all this biomass, that these different species of often white rot fungi are basically growing on. It can be basically, well, oftentimes we just be dumped. So it's a great way you can basically pick it up. Oftentimes it's a great resource to work as well. Next slide. So even some examples of how you can basically have these beds laid out. Some example of basically kind of just letting it sit and can decompose over time. Basically, in general, this mycelium going through the substrate is going to be a food source for a number of microarthropods and even nematodes as well. Going through the aspect of making sure when you lay it down, oftentimes if it's just kind of laid down on the outside, often a block, you're better off if you're taking it. You can go through the process of covering it with some kind of mulch to basically allow it to decompose. We understand fungi. Fungi, they like to be covered. They're mostly water. Most mycelium is clear. They don't really have the pigmentation to survive. But we can understand here some of this aged mushroom substrate. We dig it up. We start to see a darker brown color where the top layer of the biomass may be kind of lighter. The underneath it is darker. Also, too, it's a great addition to worm compost. There's plenty of biomass, that basically in there, and also lots of microorganisms growing on and in the surfaces, which the worms are actually ultimately consuming because they're going to be attracted to the microbiology, which is acting to decompose and cycle these nutrients. Next slide.
Let me interject that, on this slide, on the left, this is just the mushroom substrate that's been sitting there for a while. And this was never inoculated with red wiggler worms. It was just left on a concrete slab. And then after like 5-6 months, the worms found their way into it and started composting it. So this is a material that these organisms are drawn to naturally.
So even further, even when talking about the spent mushroom substrate, this is something that you can also continually expand as well. So, when it comes, so this is something even too when I demonstrate, when I do teach about fungi, a huge part is because there's kind of a gap in a lot of education when it comes to fungi. I think back to my, in college, my intro biology lab class, we got a packet. We were handed it with the, kind of going over all the different kingdoms of life. Every kingdom had a page and a half description. The intro section, fungi had a paragraph. So it's very important kind of with teaching these different examples of how, like, getting a sense of what fungi are and how they can look on the macro scale. Obviously, when you're doing soil microscopy, investing in soil food web, kind of seeing that, you can basically go through the process here. So the previous slide was some mycelium growing cardboard, that I inoculated. And the next slide is pretty much that same cardboard that was rolled up and then let's sit in a container over a couple weeks, and we see this mat of mycelium has enveloped the cardboard, but also grown out as a film over the plastic container. This organism is basically trying to explore in its environment and survive, and we see little bits of exudates on the tips as well. This organism is producing it as a means of its digestive enzymes, but also antimicrobial, antiviral, antifungal compounds to protect itself, because they're basically putting out these guttation events. Fungi digest then ingest, we ingest then digest. But the reality too, even from spent mushroom substrate, you can pretty much grow it out on even further, fungal foods. And basically, I think about this even too is with sheet mulch. Next slide. Even with aspects of sheet mulch, you take sheet mulch, you layer it out. If you're pre-inoculating with mycelium ahead of time and you're covering it, this is a great way. It's going to basically allowing the microbiology in this wet, moist environment, obviously not too wet, but maintain the right temperature. It's going to allow it to break down and decompose. This is a slide example when I was working on this mycology apprenticeship with this now probably called Beam Center, teaching students about fungi, but also the roles of soil through hands-on applications. The first day we had pizza. It's super common in New York City, big group event. The problem is though, after you're done, you have the pizza boxes, which, isn't too bad. We're very lucky in New York. We have a paper mill, which takes our cardboard and turns it back into paper. However, one kind of thing is that any of that paper is covered in grease. That's kind of a problem. Oftentimes you can't paper recycle the cardboard or material that's covered in the yeast in grease, because pretty much it'll act as food for microorganisms. It might make a biofilm in the actual machinery, and that's caused downtime. So oftentimes any paper biomass that has an organic material on is often thrown out. However, fungi not only have the enzymes to break down the cellulose and lignin, but also they have lipases. They have the enzymes that can break down fats. So we took all of our pizza cardboard, we shredded it up, we threw it in a big Starlight tote. We pasteurized it with boiling hot water, and then we inoculated a couple blocks of mycelium. And the whole point here wasn't try to grow mushrooms, but to basically demonstrate to them that you can pretty much cultivate this this biodigester, basically turning into this fungal stomach in a bucket. It's a nice little rhyme off the tail. Next slide. But even further, you see these little bits of white pelly mycelium, but also these bigger kind of clumps, and also these little pools of amber. So we're seeing the mycelium itself grow in, on, and through the cardboard. And then we see these little pools of amber. These are the exudates. These are the materials. These are the exudates that the fungus is pumping out to protect itself, digest its food, but also inhibit competition as well. And then next slide. And then we also see these primordia. We see these structures. This basically the fungus getting ready to produce mushrooms. Now, in this bin, one of the limiters for mushroom size is predominantly the amount of nitrogen that is available in the substrate. Oftentimes in mushroom cultivation, you'll have an aspect of 50-50 where they'll try to get a balance of 50% of high carbon nutrient in the substrate and a high nitrogen composition in the substrate. So that way you'll maximize the amount that you can get maximum fruit body growth, but you're not going to let it potentially kind of go moldy or get contaminated in the production structure. But the whole point is to demonstrate the fungus is attempting to make fruiting bodies, but really limited by its ability of its nitrogen source. But we're just trying to cultivate mycelium here. So even with spent material, you can go on and basically produce it. Next slide. But just demonstrating what would happen if you were going to be inoculating and you see more primordia. Next slide. But yeah, an example here, even what it looks like at home, you can get a Starlight tote. Basically, in general, you basically take your cardboard from a week, shred it up. I recommend cutting it with scissors is a pain in the butt. Tear it up in a range of sizes. You don't want it too fine. It'll go anaerobic in pool water. Maybe a mix of fine shreds to big clunks. Pasteurize it with boiling hot water and dump in some spent spawn blocks. And then basically once a week, check on it, feed it some new cardboard. And at the end of the month, remove a third of it and then pretty much add it in. You'll keep it going. The one thing to note is definitely keep in the place that is definitely kind of keep it into place. Don't let it level too high. if you fill it up too high, it's better to kind of go for a shallower container. If you get any kind of distance beyond a foot, you'll get a lower oxygen gradient, and that can kind of tend to lead to more mold. And definitely there's been times where I was kind of working in a space where someone was working one of these and they had a cloud of trichoderma spores erupt of it after they kind of went to turn it. So on that note, trichoderma is kind of known as the mushroom cultivator's bane. It is a mycoparasite, so it's pretty much a fungus that basically parasitizes other types of fungi. However, it plays a critically important role. Definitely heard you can buy a trichoderma inoculum for different soils, but it's all about balance. And even some of these blocks from mushroom compost in general, or not mushroom compost, but spent mushroom substrate. When they're aged, the variety of diversity you get, where it shifts from being a monoculture to a polyculture, can actually yield to that diversity over time. However, it's all about diversity, all about balance, and ensuring aeration in general. But this is something that does show up commonly on mushroom cultivator blocks, and oftentimes some mushroom cultivators, they do know that some trichoderma does run away inside of a bag where they fail to kind of maintain a monoculture inside of their production facility. They'll just kind of abort the bag and dump it out back as a measure as well. But an aspect that really it's these organisms are trying to take basically these organic materials and break them down further and turn them from a kind of a disturbed system, like a primary, basically a bare material, like kind of that primary stage of succession, to more advanced succession over time.
All right, now we're going to move away from the world of mushroom cultivation and mushroom farms and get to the real diversity, because all the stuff we've been talking about is in those last two techniques. Even with the spent substrate, we might get the trichodermas, and an important point, there's a lot of different species in the genus Trichoderma, so they're not all the same or doing all the same stuff, but we're still looking at kind of a more narrow range of species to work with. And probably some of the more potent tools for working with fungi involve trying to get more of a natural wild diversity, which has been a theme of a lot of the talks going on here. And on that note, we're going to talk about mulch and organic matter as a tool for managing your fungi. Now, there's quite a few reasons why mulch is good for fungi. First off, it's a vector, so it's fungus and other microbes travel on mulch, so inoculum, vector, they get from one place to the other. In addition to being a vector, it's also a food source, so you're going to be able to feed the fungi and the microbes with your mulch. And then mulch also helps maintain climates that are conducive to fungal growth, creating microclimates with more humidity, darkness, insulation from temperature. These are things that will make your saprobes want to grow better, because they need fresh air, they need moisture. If they dry out, then they stop growing. That's pretty much how most fungi work. So creating that microhabitat, and additionally preventing compaction and maintaining that soil structure, because if your soil is uncovered for any amount of time and either something steps on it or it gets rained on, even if it gets watered, all that is just going to push down the soil structure, and then the tunnels and the pathways between the aggregates will be smaller and smaller, and then your saprobes and other beneficial microbes won't be able to grow in it very well. Now, certain types of mulches are more fungal friendly. A few examples is we have the leaf mold, and just in general, the more you shred it up, the better it is for it. Straw and hay are usually pretty good for fungi, but they tend to have seeds attached to them, which can be a little problematic depending on what your growing practices are, and if you don't want unintended grasses growing out of things. And then wood chips are sort of the go-to fungal mulch. If you're trying to use them for propagating microbial biodiversity, you want to be aware of maybe avoiding mulches that from woods that have a lot of antifungal and antibiotic properties, such as pine or cherry and locust, and things like eucalyptus and walnut that are allelopathic and actually produce toxins to poison like the competitors and things like that and prevent them from being decomposed. So don't use those things. I don't know what the big trees are that are available in your region. They're good hardwoods. Here in the southeast where I live, it's we're usually looking for oaks and maples and poplars and things like that. But talk about leaves for a little bit before we get into the wood chips. And this is a study published recently that's part of a more and more research that's getting into this topic of how fungi move through different parts of the environment and potentially different life cycle phases. And what this graph shows on the right is the overlap between, they're surveying the fungi that live in the leaf litter soil and the decomposer saprobes that live in the leaf litter, and then the fungal endophytes that live in the tree leaves growing above the litter. And what they found is that if you look at the upper right, from just DNA, which is just kind of analyzing who's there, it's about 42% of the fungi they identified in the leaf litter, they also identified in the plant leaves as endophytes. So that's, 40% of the species here are coming from the actual tree leaves, and that's how they're getting down there, probably. More than likely. It's hard to tell specifically with this. But what's more interesting is the bottom right graph, which is the RNA-based analysis, which is more judging who's actively decomposing the litter. And when you look at it from the RNA component, it's actually the fungi that existed in the plant leaves are doing a majority of the decomposition. So the whole point and reason I guess I'm bringing this up is if we're using leaf as a mulch, if we're trying to deal with leaf mold practices, think about that we're creating like a bioreactor and we're harnessing all these fungi and other things that are growing in the plant leaves, as well as the ones in the soil that we put the leaves on top of. So here's a leaf mold pile, and I put mold in quotes because molds are usually viewed as fungi that don't form mushrooms. And we do have mushrooms here. But if you look at the middle of the pile, it's turning into a kind of pretty nice composted dark brown material, eventually going to make its way into soil. But yeah, we got the mushroom there. We got all this mycelium growing over it. And this is all purely just rake up the leaves in the fall, put them through a shredder, and then leave them in a pile and walk away. And it turns into this inoculum that has, different types of mushrooms, including the bluet, which is a gourmet edible mushroom, which is also a leaf litter decomposer. And so this was growing in the pile, not inoculated. It just came in from the soil somehow and is fruited for multiple years in a row. And, on the right, we can see the close-up of the blue. It's kind of got this like soft, fluffy cotton candy looking mycelium. But so this is one type of mulch that really will improve the fungal content of your soil if it grows out this way. We also, there's wood chips are probably more commonly thought of for fungal mulch. As a project I was involved in a couple years ago with a rural county landfill. We were testing laying down wood chips on their landfill slopes to reduce erosion and improve soil quality. And we tried out, like, what if we add some bags of mushroom spawn to the wood chips and like grow it out on it? Maybe that'll hold it together more and make it more stable, prevent erosion a little more. And so we set up these piles of wood chips about a foot, 12 to 18 inches deep, inoculated with some straw on top. These are what the test plots looked like on this hill slope. At the end of the study, went and cut open these plots and looked on the inside to see if the mycelium was growing all right. And we found quite a bit of this nice white filamentous structure. But the interesting thing is that, it wasn't fully colonized. It was maybe growing on like 75-80% of the wood chips. But we didn't notice a difference between the plots where we had added fungal spawn, we had actively added mycelium, and ones where we just laid down wood chips and didn't add anything. The ones that were the controls, where we did not add the fungus, tended to have as much, if not more, mycelium growing in them. So it really was it was the wild fungus that was taking off. And so, maybe we could have saved money by not buying all that fungal spawn, but then we were able to figure out, if you just stack the wood chips the right way, you're going to probably get that fungal diversity benefit anyways. And we found more than just mycelium. There's this like weird looking orange octahedral honeycomb slime mold thing growing in there. So you never know what biology starts doing once you give it a home to do that type of stuff. But so kind of some of the takeaway points here, because mulch, it's a super complicated topic. There's a whole lot to it. I think there will be a presentation on it in the coming months in this series on living mulches, which is kind of what this is a reference to. But yeah, takeaway points is if we're using natural mulch materials, your mulch already has fungus inside of it. So just this mulch just inherently is going to be inoculating your soil and feeding other microbes in it. If you're working with specifically more with leaves and stalks, but even with, if you can grind up wood, the more you can shred it up, the more you're going to avoid getting anaerobia and, water pooling up, as long as it's not too small. But yeah, it's really a big part of it is maintaining your mulch piles in a way that is conducive to this like aerobic saprobe fungal growth. So some of the main things is not having it be too big, because once a pile, whether it's wood chips or leaves or whatever, once the pile gets to be 3, 4, 6, 10 feet high, it's so big it's probably going to go anaerobic in the bottom of it, and it's going to get dominated by actinobacteria. You're not going to get the aerobic saprobe diversity in it. So, not to say every mulch pile you ever make needs to be these dimensions, but if you can actively think, are we going to try to make this mulch a really good living mulch, and then make your pile a size that's conducive to that, so that you can maintain the airflow and the moisture content and all that stuff. So these type of mulch and inoculums potentially way more biodiverse.
You got some feedback coming through.
Indigenous microorganisms in it. So another thing that helps with maintaining soil fungi, I'm not going to go that much into it because you all probably are pretty familiar with it already, which is cover crops, just having roots in the ground. Same concept with the mulch of the soil fungi habitat. You're providing more moisture, shade, and insulation, preventing compaction. And additionally with the cover crops, root exudates from the plants feeding the fungi and other microbes in the soil. So really, any soil you're growing in is ideal to the very least. Yeah.
Sorry to cut you off, but your microphone is the quality is slowly deteriorating here. It's been just getting really raspy.
Okay. Still raspy right now.
Yeah, it's echoing. It's gotta.
Hold on a second. Does it have the feedback in it now?
Oh, I think it's much better now.
Okay, yeah, I think my heater went on, my HVAC turned on or something, and there's a humming in the background. But
great.
All right. Should I back up at all, or just keep going from here?
Maybe back up just like 30 seconds or so.
Okay, right on. Well, yeah, just to recap what we were saying about the mulch. Natural mulches have fungi in them already, so you want to stack them and set them up in the proper way, and then you can get those organisms to grow. So yeah, don't make your mulch piles too big, or else the bottom will go anaerobic. That's main takeaway. Are you all still hearing me?
Yeah, you're good, Leif. You sound great.
All right. Cover crops, like I said, you all probably are already familiar with them, but they help provide that fungal habitat. The roots feed the fungi. So basically, your soil is going to do, it's going to maintain its structure and its livelihood the more you keep it covered. It's ideal not to have your soil be uncovered when you're not using it, either very least with some mulch, but if not mulch, then some sort of plant roots inside of it. All right. Yeah, so we've been alluding to compost a lot. We've written up some, the basic compost parameters here, but it's way more complex. It goes in way more deep than that. But something that Craig mentioned earlier, which is really important with fungi and compost, is to not let your pile get too hot. Chris Trump was mentioning this last weekend with IMO about don't let it go above 120 because you'll start killing off the beneficial microbes. If you're doing thermophilic composting, try not to let it get up super hot, 131, but you might have to hold it longer. That's because we're basically trying to have it be a habitat that these fungi won't get, too torched and need to go dormant in afterwards. So yeah, this is a mushroom compost pile, right? It's all mushroom substrate, and it looks pretty. We got a lot of mushrooms to grow off of it, but this is actually this is a terrible compost pile. It's a terrible recipe because it's all the same thing. So it's too brown. It doesn't have enough nitrogen on it. It's not going to cook. But you can still use spent mushroom substrate. But if it's maybe 60% spent mushroom substrate with some manure and then some grass clippings, other things, actually can turn into pretty great compost. But beyond, we've spent enough time talking about spent mushroom substrate and mushroom blocks. So let's talk a little bit more about how to promote the natural wild fungal diversity in your compost pile. I mean, a big part of it is fungal foods, inoculating your pile with fungi in some way as well. Then, we have some of the fungal foods we think of more because they're hard, kind of woody plant material. So bacteria and other organisms cannot actually decompose them. So that's why they're good fungal food. But fungi also like oils and fats and proteins and stuff, which is why sometimes things like fish hydrolysate and oats and things like that are recommended as fungal foods. But a little tip, a little trick you can use to boost the fungal to bacteria ratio and the fungal content of your compost is once it's been cooked and it's fully finished, it's completely done, and it's cooling off and curing, then add some more woody fungal foods at that point, because then you're going to kind of favor the organisms that can break down that wood and potentially heighten the fungal populations. So that's about that for that. Are you all still hearing me?
Yes. Can you hear me?
Yeah. Is Craig still here? I don't see his video anymore.
I think he might have fallen off a little, but
all right. Well, right on. Well, I mean, we're wrapping up here. We only had one more slide or so that he was going to do, which was, yes, natural farming is a great way to improve fungal content of your soil. I'm not really going to talk about how to do it. You can look up, watch Chris's talk, or go look up his work. There's a lot of people who are doing a lot of good work with natural farming who know far more about than I do. But the premise is the same. A lot of the similar concepts I was talking about, the living mulch and the leaf mold piles, is where we're trying to figure out what's already living here and how can we grow those things out and use that as a soil inoculum. So if you look, there was kind of a sequence in the management strategy you went through where we started with talking about how to take one specific decomposer fungus and get it into your soil, but then radiating out from there, and how do we get multiple types of fungi and multiple diverse types of microbes and get them into your soil. Because even though this talk was about saprobes and yeasts and kind of these fungi doing their nutrient cycling, hopefully we've demonstrated that it's not the fungi operating on their own. There's all this synergism and symbiosis with the bacteria, the plants, the protozoas, all the organisms. So diversity is the key. We want to promote diversity and inclusion initiatives for our soil microbes so we can get them all in there and all saying their thing and doing their part. So here's the references for the talk. If you want to look these up, maybe screenshot them on the replay, and or you can contact one of us. But this is all the scientific literature that we referenced throughout the talk. So we didn't make it all up, not most of it. But anyways, thanks for listening, and I guess we got time. We have about 30 minutes for questions now. If you want to get in touch with me, my company is Integrated Land Enhancement. Go to the website or send me an email at mycoscapes@gmail.com if you want to ask about any of this stuff or whatever comments. And you can reach Craig at nyc.nyc. Follow him on Instagram at nyc.nyc. Get the N's and the M's correct. But and stay tuned. We're actually going to be releasing a show going real in depth on a lot of these applied mycology topics. Maybe not necessarily for growing plants specifically, but different topics. So keep an eye out for that if any of this stuff interests you. And and look who's back just in time for the
Q&A. We love these last minute comebacks. All right, so we've got a pretty good list of questions going. You guys maybe can just rapid fire some of these. Some of these are a little more in depth than others.
All right.
So the first one we have here, and again, guys, for everybody listening, you get to upvote the questions. This is a little bit different than last week. So if there's a certain question that you like, please go through and upvote that, and that's going to obviously populate to the top so that we get the best questions answered. That's a pretty cool feature that this platform has. So Sean R. has the top question. I have seen several indoor raised bed medicinal grows that utilize a thin fungal mat that spreads from each edge of the bed. The layer is fairly thin and created by mulch. Do you believe there are any positive or negative effects in doing this?
I think it would probably be a positive effect because you're increasing the fungal diversity, but also if you're creating that thin mat, it's probably going to be having structural benefits too, because the mycelial mat is going to help hold more water, improve soil structure. In terms of potential drawbacks, it seems like it would generally be a positive thing to do. Always something could go wrong if like there was a pathogenic mold that took over the mat, or if the mat is too dense and didn't allow water or air to get through. But generally, it seems like it would be a beneficial thing. Yeah, because it's kind of like we're talking about with the inoculating either with the living mulch or with the mushroom substrate into a raised bed and kind of kickstarting that activity. Not necessarily because we're trying to grow that species of fungus, but there's more fungi, more things can eat the fungi, and the cycle of life goes on.
Can you guys hear me? I had to dial in my audio. My computer is out, but I'm here for the Q&A.
All right, you're back.
Right. Yeah, and we can hear you loud and clear.
Excellent.
All right, so this next question is from Phoebe. What is your recommendation to supplement the soil with manganese or magnesium? Or magnesium.
I would say both.
Yeah,
right?
you want to start? Yeah, go ahead and start, Leif. I'll follow.
I was just saying, like, if it's an either or, I think it's both because both of those are necessary. But magnesium has more properties with like soil structure and flocculation and things like that, whereas manganese, I think, is more just about providing a trace mineral to help with like special enzymes in plants and fungi.
Yeah, definitely. And when it comes to concern about like how to supplement, the factor is once you're getting up the biology at that point, which you're playing a role, what we're able to extract, those micronutrients, macronutrients, and trace elements out of the soil parent material, but also the organic matter. So the reality in general is application. Adding too much, I think as Pericles said, the difference between poison and medicine is the dosage. I think in general with over application of these materials, which has kind of been the product of the chemical era, can be complications. So I wouldn't say focus more on the biology and the organic matter. That's kind of one of the biggest aspects to it.
Clarification, it was Paracelsus. It was the father of modern toxicology. The dose makes the poison is his famous quote.
Well said. Sean R., when we apply lactobacillus, what determines when the bacteria will make lactic acid, or is that acetic acid?
Lactobacillus, they produce acetic acid.
Acetic acid.
That's often, there's been times where I've been presenting where I sometimes say, because you say lactobacillus, you think lactic acid, but they're actually producing acetic acid.
All right. Sean R. with another one. On the average, does trichoderma consume mycorrhizal faster than the mycorrhizal can reproduce or fight?
That's a good question. I don't know if there's a clear defined answer to that, because first off, there's different species of trichoderma that vary in their virulence. I'd imagine that it could happen, especially if you don't have diverse soil biology and the trichoderma is like running away as the dominant organism, which when we see in the trichoderma growing on the spent mushroom blocks and things like that, it's taking over because it's like the thing that's best suited for that. And there's not other things that would be eating and consuming the trichoderma. So I think it probably could, be problematic to mycorrhizae, but if it's a diverse enough soil ecosystem, something will probably keep the trichoderma in check.
Yeah, and also too, it's the factor is when you're buying a bag of spores That's an ungodly amount of concentration. It's kind of like there was kind of a bit of a joke, like, if you had rival mushroom cultivators, you could ruin their operation with spores. But, the reality of what they're being produced in a natural environment, if there's like an abundance of biology kind of playing that role where, when you move more towards biodiversity, there is basically this kind of out competition in other kind of roles where they kind of pick up those different trophic levels and inhibition. Some of the interesting stuff is thinking about maybe some of the roles that, when you have an abundance of different microbes, certain types of fungal pathogens, like, I forget the name of the top of my head. Fusarium can oftentimes be changed over from being a pathogen to kind of being, not just a sapro, but also mutualist by certain species of bacteria like Pseudomonas. So it's kind of the fact is we're just kind of scratching like the surface of kind of these different roles, metabolic roles. It's kind of an aspect of science where I'm kind of getting into looking at programs where you're able to do the microbial modeling, but also the metabolic modeling. So that's kind of the more applications of like computer science and neural networks and, some of these deep kind of machine thinking into kind of these biological systems, which are the only way to really approach it at a scale and a sensible time frame. Nothing like some good old soil microscopy, but the world does go deep.
So you're basically saying that to truly know the answer to this question, we're going to need like machine learning AI algorithms of metagenomic data, because otherwise it's kind of a little too hard to observe.
For a definitive answer, but I think for day to day, just move more towards diversity. That's kind of what the larger trends say.
Okay. Yeah.
That's the real beauty of this for people that are more of self-taught kind of thing. Is it really is diversity. Once you kind of you listen to a lot of these talks. A lot of that stuff just comes down to the diversity that you're able to achieve. From Julie D, is cedar wood good for fungi?
I think it is not. I don't think cedar is I think cedar has pretty good antifungal properties in its wood and its sap. It's not one you would necessarily want to use for promoting saprobe of fungi growth.
I second that. Yep. Sorry.
Yeah. Oh yeah, and tying that in, it's actually, I think, when it comes to tying it into natural farming, when you're doing collections, you kind of want to aim for something that has a really high carbon nitrogen ratio, like cedar in general. So it's not just the carbon ratio, which makes it from bacterial breakdown, but also too the structure is the various kind of antifungal compounds that are in the wood as well that will kind of hold it up. So oftentimes the notion is that you have to let those things significantly off gas and sit before those volatiles would basically would still be stable, which would impact fungal growth.
That is well said. All right, Phoebe, what are good sources endemic to Northern California that are abundant with manganese or magnesium?
Endemic. That's I don't know if I can answer that. Usually I would use trace mineral fertilizers like azomite or sea minerals, but those aren't from California. The sea minerals are from the Baja Sea, so that's more of northern Mexico. Because a lot of this stuff is coming from like ash or rock or salts or things like that. So I don't know if any of those are specifically in Northern California.
Don't a lot of the quality seaweed and stuff, isn't that all Icelandic, or is that all marketing?
It could be. Yeah, I'm not sure. I don't know about like the best sources for seaweed or kelp, but they do have a lot of those minerals in them.
Yeah, I don't know if this is true, but I remember on the forums they were talking about how kelp had about 20 or 62 trace minerals. So there's a lot of bro science from where I've come. So again, Leif, and we really appreciate this because it's hard to get a lot of these questions answered from people of your knowledge base.
Yeah, and the challenging thing with a lot of these types of questions is there's not always straightforward, easy answers to them. Our collective human knowledge isn't at a point where we can precisely answer a lot of this stuff. So it's kind of like you got to work with, what do we know, and then kind of some amount of estimating and guesswork. Kelp has a lot. Most things that come from the ocean have a lot. There's a whole theory about why things from the ocean are so good as trace mineral fertilizers, and it ties into the fact that most marine life has the same trace mineral balance in its body. Like there's not a huge, in terms of the balance of whatever the manganese and the the magnesium and the boron, all that. In marine life, it's a pretty similar ratio in almost all animals, and it's very similar to the ratio that's actually in the ocean water, which it's not the same everywhere in the ocean, but it's relatively similar. But once we move on to the land and we're looking at soil and plants and terrestrial animals, all of a sudden those trace mineral ratios are way different. And there's a lot of speculation that really healthy soil would have a similar ratios as the ocean, because it's kind of the ideal balance for life. But throughout time, with erosion, development, land use, deforestation, the minerals leach out of the soil. And then that's why a lot of the soils are way more depleted in these things. So the idea is like the ocean has the balance, the minerals washed off into the ocean. If we can put the ocean back into the soil, then we can balance the minerals. That's very vague and abstract, but it's kind of like a nice idea to think about.
Craig, do you have anything to add to that?
Yeah, and kind of in general is that we have to understand that life had to become adapted to the earth. Terrestrialization is the process by which most things are roughly 70% water. Most organisms, whether you're a human, an animal, a bacteria, maybe a bit more as some of the types of microbes. But the reality in general is you think about that in the ocean, everything's kind of in solution. It's an aqueous environment. Versus in general, you have to understand once you're a terrestrial organism, you're like a sack of seawater, basically, in that aspect. So the reality is to kind of balance your mineral nutrients, you basically have to eat plants, terrestrial plants, or you can even eat aquatic plants, or eat terrestrial or aquatic animals that eat terrestrial or aquatic plants in that capacity. So that's kind of the whole aspect of understanding that, life had to adapt to live on land, whereas some species went on to land and then moved back into the ocean. So that's kind of to think about that as well. Understand that the ocean is one giant solution. And it's really hard to understanding, think about this, okay, let's say you wanted to measure the concentration of nutrients in one part of the ocean. It's okay, if you're sitting in a rowboat in a pond and you took if you say, I'm going to take one bucket of water out from this part of the pond, and you're holding one bucket of water, did you do it? Because it's kind of just all amorphous. But the kind of general notion is that it's a solution with minerals and nutrients. So think about corals, right? Corals are able to pull the calcium out to build their skeletons out of the solution that's in the water itself. It's a bit harder to do that when you're kind of living on land and you need to wick that up from somewhere else.
Although lichens do that on land. The lichens are fixing nutrients out of the air and incorporating them into their body. And then when they fall off the trees, it ends up in the soil. So it's another example of this nutrient fixing going on in nature. But don't go pick lichens and amend your soil with them. That would be very ecologically irresponsible.
Yeah.
Our next question is from Clay. Are there any known cultivated yeast that produce jasmonic acid?
Yeah, I couldn't tell from what I read if any of the yeast actually specifically produce jasmonic acid. I think it's more so that the yeast interact with the plants and then they cause the plants to produce jasmonic acid. I don't have the names of the specific species or know if they're cultivars, but it's something that's also observed with mycorrhizal fungi. That it's they're actually telling the plant to change its salicylic acid, jasmonic acid production. They're not actually doing it themselves. So it's kind of they're more whispering something in the plant's ear to encourage it. But yeah, sorry, I don't know of any specific cultivars that do that.
Craig?
Similar answer, but kind of going off what Leif was saying about those interactions. Those kind of dynamic interactions between macroorganisms and microorganisms, between whether they're plants, fungi, bacteria, kind of thinking about where you have modification of expression. It's one analogy I kind of give is you think of yourself as a singular entity, like a human being, but you're 35 trillion cells that are 100 different cell types that are all communicating in different ways. They all have the same DNA, but they're only expressing a certain part of the genome to function. So that's kind of the thing to kind of understand is these interactions could be influencing production of different things based upon the environment, stressors, and also who's home as well.
Next question is from Sean. Any opinions if manure, leaf mold, or mushroom substrate produces a superior vermicast?
I like you're talking about like worm compost, right? Like for like what's the best
worm casing
ingredient for worm castings? I'd say it's probably mushroom substrate because worms. I mean, when worms are like your red wigglers are going through your worm bin, they're eating the fungal mycelium and the bacterial bodies. That's their actual food source. So if you have spent mushroom substrate, although I will say straight up spent mushroom substrate isn't the best because it can be a little dry and the residual sawdust from it can be a little like abrasive for the worms. So sometimes it's nice to mix something else in. But like the fact that it's covered in fungal mycelium makes it really good worm food. Whereas leaf mold could be, but I almost feel like leaf mold is already a good inoculum on its own and it doesn't really need to be composted. So I would probably just use it as it is. And then manure, I don't know. I've never put manure in a worm bin. I remember hearing that it's not good. It'll start rotting and you'll start attracting, like other types of insects, but I haven't tried it before. But I'd say, yeah, spent mushroom substrate is one of the best compost and vermicompost ingredients that there is. If you can, if you can get in touch with a mushroom farm that's trying to get rid of it, it's great for, decomposing and feeding.
And kind of speaking to like the water content, usually worms earthworms tend to like that range between 70 to 80% moisture content. Usually 80% is where they reproduce. Once you go past that, it gets anaerobic and they'll just kind of peace out. So, I guess depending just kind of in principle, whether you're working with the leaf, whether you're working with the mushrooms substrate or manure, it depends on how dry or wet that is. For example, like you could have road apples or like rabbit pellet manure where it's far drier than wetter versus like different sources. So that's kind of another aspect. Personally, don't anecdote, but I think definitely the hydration factor plays an important role.
I personally have been playing around with the manures, cold manure from a lionhead rabbit, and I've started to notice a lot of fungal growth from that. I would imagine also another manure, and I hope you guys would co-sign on this if you think this is true, that alpaca manure would probably be the most sought after of the manure to try to really build up those fungal bodies in your vermicompost.
I hear great things about alpaca manure, but yeah, like I said, I'm not that knowledgeable on which manures benefit fungal growth the most. But I hear good things about alpaca manure, and they all poop in the same place. Both rabbits
and alpacas are fluffy, so that might be something with it. I don't know. Yeah. Mm-hmm.
The beauty is, like you were just saying, the alpacas are in a way kind of like dogs. They'll be able to poop in one area. It's easier to clean up. And the same thing at least with the lionhead rabbits, you're able to potty train those. So it does make a big difference than just using like a white dwarf or something like that. So I do feel like species matter with that, but you are able to achieve.
Also the diet, if you're getting manure, you want to, unless you're seeing over those animals and making sure they're getting fed, food that they evolved to eat. And, that's one thing about it. If you're just picking up random manure and you don't know what the diet is, that can definitely have some downstream effects that you can't really control, especially with salt content, pathogens, other things like that. So that's another thing to keep in mind.
Yeah, herbicides are a big thing too. I've talked to people that like completely destroyed their garden because they picked up horse manure to mix in their compost, and the farmer had fed their horses grains that was covered in 2,4-D, which is an herbicide that was used in Agent Orange. And it was in the manure. It didn't get biodegraded, and then they made compost with it, put it on their garden beds, and it just killed all the biological life in their soil because the herbicide was still in it. So yeah, being aware of the diet of the animal's manure that you're using is pretty important.
This next question is from Ron. Do you have data on antifungal and I don't know that word, gentlemen, pathic properties of different woods? Some who looked into this discount the effect. Sorry, guys, I don't know what he's talking. Can you see that question?
Yeah. I think I understood the question. And I mean, I don't have any data on that. I'm basing it more off of, stuff I've heard, and also my own observations of trying to inoculate things like pine wood chips and cherry wood chips with fungi, and they never grow on it when I do. But that is anecdotal. In terms of the allelopathic component, and whether it's with walnut, eucalyptus, if that's preventing it, that was more of a educated guess. But I don't have any research demonstrating that. But I will say that I've tried inoculating pine and cherry wood chips quite a few times, and it doesn't really go that well usually.
Similar, yeah. It's just, yeah.
Are there any certain, like you mentioned before in this talk, the oatmeal seemed to be for me something that was able to kind of especially if the soil was already kind of alive and thriving, was a little bit of that catalyst to kind of speed things up, maybe as a farmer kind of improve on Mother Nature a little bit by adding that oatmeal.
Mm-hmm. Yeah, I could see that, because the oats and any large kernel grains, it's packed with all sorts of proteins and nutrients that all the microbes love, including the fungi.
Yeah, I feel like a lot of us were using that in our gardens. We just didn't really know what was going on. The first time that I experienced that, I threw all of it out. Same thing the first time I ever got springtails, I threw all of that out. So, to me, that's throwing away time. But, both of those, especially leaf litter. I mean, if you guys can get leaf litter from a friend, in my opinion, that's kind of like capturing time or getting time from someone, because that's going to be able to inoculate things on a much quicker basis.
Mm-hmm. Yeah, and definitely, I think part of it too is the view that, definitely seeing other biology than what you want. The notion that when you grow a monoculture, anything that's there is like a pest or a pathogen, where it's like, no, there's more nuance to it, right? It's like you might see like a springtail, think, oh no, it's like a thrip or something that's in like a aphid or something. But you got to break out your jeweler's loop and get down and roll with the bugs and realize, oh wait, no, they're playing a much bigger role in the nutrient cycling.
Well, one of the biggest issues that I had, and I think it's still out there, is if you Google springtails, the first, I would say, 20 things that come up, it's all of a negative tone, how to get rid of springtails. So I feel like a lot of us out there had to learn the whole hard way, because when those springtails, especially when you would first start watering and once they would really start to develop, it would look like it snowed like Christmas morning once you start to really water that in there.
Yeah, and that point you made about springtails and looking them up is very much true of when you look up fungi a lot of the times too. The best knowledge we have about what fungi actually do is all based on studying pathogens, because that's where the focus has been. Which is why this how to leverage saprobes in a positive way there's the scientific literature is pretty sparse on it, in terms of taking it out into the soil as a growing method, because people haven't really thought of doing that. And that was also what I read with getting into the environmental roles of yeasts and yeast being involved in nutrient cycling. That's another topic where it is it's hard to find anything on that topic before the year 2000. And some of the initial studies that are getting into yes, the yeast, solubilize the minerals and produce the ammonia and this and that, but they always begin with soil yeasts are very poorly studied, because it was always assumed that they weren't doing anything important. But then it's they look at them and they realize they actually are doing important things. And that's, I think, a common thread with probably a lot of these talks and people talking about these different types of soil microbes. It's a lot of these things that got overlooked because people were just it's not doing anything. Why even explore it? But then when they start to, and part of it's because we have better research tools and techniques now to actually understand what they're doing.
A real quick question that came from Instagram earlier last week. For people that don't understand natural farming or Korean natural farming, do you think it's an excellent substitute to use the kefir instead of lactobacillus to just kind of drizzle that down and start to build up more fungal aspects from that?
Yeah, I would be completely guessing when I answer this, but I'd say it probably benefits it. I know I always, have old scobies from brewing kombucha or June or something and put that in, because in a kefir, it's a SCOBY too. It's a symbiotic community of bacteria and yeast. So it's probably it's like you're adding a probiotic to your soil, basically. Now, whether those organisms survive in the soil. Yeah.
And it's something that kefir, that kefir grain, that SCOBY is basically the attempt of that community to survive in an environment that's kind of hostile. So they're that kind of mat, which are basically like what a SCOBY or a kefir grain is, like a microbial cellulose. It's pretty much this amalgam between the two. So yeah, it's something that's stable, could be added back in. You're getting that diversity as well. But it's something that like is a propagule that you can inoculate it with it, because you go on to inoculate other milk or kefir water or like, tea to further on your lineage of like SCOBY or kefir for kombucha or kefir. Yeah.
And since it's dairy free, do you guys also agree that it's going to be easier on the microbes and the whole environment? Or is that just kind of, again, bro science?
It kind of makes sense. I mean, I don't know the scientific theory necessarily, but just when you pour milk out, it's like it kind of rots and creates different types of, kind of anaerobic kind of microbes, less so than if you pour out, say, like almond milk. It's like in terms of how much it's going to turn into like a different type of microbial community. But yeah, I don't know if that's, if there's scientific evidence behind that or not. That's more of just, the smell test.
Craig, what do you think about that? Yeah.
Well, it's like understanding like what is milk. Like milk's kind of a colloid, so you're getting this this kind of culmination of water soluble and insoluble molecules tied up in fats and sugars. And there's a lot of free available food for lots of microbes, definitely bacteria. And so it just depends. It's really hard to quantify it. It's better to kind of go on the safer side, I would suppose. But kind of a notion, it's like thinking about what the first principle, what that is. And like kind of when you understand like something going rancid, right? Like I think a lot about how like if you're making a lab serum, you're washing rice, your nose is kind of that ultimate indicator, because it's such like a tied input. Like, with human beings, domesticating animals, milking them, like, we evolved to sense when something has gone foul, like beyond when we can't eat it where it would make us sick. So kind of like your nose knows in that point, but kind of thinking from a first principle, but also kind of relying upon that biological feedback you'll get.
Your nose knows. That's wisdom. I feel like that has been passed down all over these last few years, that is really taken to heart. When you first kind of smell. Like I was fortunate enough to go to Chris Trump's event where you had the hands-on experience for 5 days, and that was one of the things he loved to do, was get you to smell rancid things. So that you would never. It was almost like an imprint in your mind there for a second that you probably won't forget. The way how pungent some of those things really were.
Hopefully with Web 3.0 we'll get smell-o-vision or something. Or just like KNF scratch and sniff or something.
Yeah, your nose knows on that for sure. I feel like it's the same way with soil. Like certain consulting work we used to do, that was one of the first things, is you kind of like pull off a fan leaf and see if that's sweet, and then kind of pick up especially kind of dig in a little bit and smell that soil system. Usually, I've personally found that if I feel like I have everything managed and dialed in, that it does start to smell like a little sweeter, old growth forest type smell to it on a microcosm scale. And obviously, the goal is eventually to get to that level. But that seems where things start to turn, where it does smell sweeter.
Mm-hmm.
Yep. Mm-hmm.
All right. John M., have you ever heard of a product called Root Shield? Plus, there is 2 specific types of trichoderma that are known to help the roots in the rhizosphere and protect them from pathogens.
I haven't heard of that product. No, have you, Craig?
I've heard of it before. I've definitely stumbled across it. Like some of the things I do follow are different inoculums. When it comes like any inoculum, it's part of it is like understanding how they're going to go through the process of isolating and classifying them. And, so it's something that like they might have a really good process and like comes down to that, but it's kind of hard to really dial that in unless you know someone that can assess it. But I don't want to like say, don't go for it. But the reality is, there are some species of trichoderma that can be beneficial, but there are also some species that can be pathogenic, but also some ones that will kind of regulate and keep into the udder. So it's really hard to like kind of point a finger to it in general. So I'd really say it comes up to the anecdote of what your experience is, if you had good prior success with it. So
there are some studies, I can't remember the species used off the top of my head, but studies where they are like inoculating a tomato plant roots with a certain species of trichoderma, and then the one that got the inoculation grows bigger and has nicer fruits and stuff. It might have been Trichoderma harzanium, but don't quote me on that. But yeah, so there's research kind of showing that different types of trichoderma can break down pollutants and potentially stimulate plant growth. But yeah, I don't remember the species off the top of my head.
I feel from experience, I mean, this is just experience, but if you are adding trichoderma on purpose and you're growing cannabis specifically, you're shooting yourself in the foot. All right, Sean R., do you have any stories of diatoms used in conjunction with mushrooms to remediate soil? Good question.
No, I wish I did.
Not with diatoms. I know that Leighton has a couple stories of working with some Pseudomonas species. He mentioned this one time he had to do a remediation job on an old, it was a gas station where they had all this, these tanks that were seeping stuff out. So it's another story time for Leighton. Not for diatoms, but there definitely are microbes that do have the capacity to really decompose some really gnarly organic contaminants.
Yeah, and I could see how diatoms would synergistically interact with the fungi because they tend to be a nutrient-rich material that the fungus could be utilizing to better degrade stuff. Because a lot of the issue with the bioremediation of soil with fungi is figuring out how to get the fungus that can break down the contaminants that actually grow in the soil. Often involves adding co-substrates like sawdust or things like that, but then it's trying to compete with the native microbes. And then when we're talking about the enzyme degradation systems that need the trace minerals. So yeah, I think feeding other, good nutrient inputs to the fungus for that purpose is probably really important. And ultimately, mycoremediation of, trying to use fungi to clean up the soil is going to work best with a diversity of probably fungi and bacteria and other organisms. Because the same way that we're talking about how the fungi will break down, some break down the white rot lignin bonds, and then some break down the smaller bonds, and then bacteria break down even smaller bonds. Those concepts still apply when we're talking about petroleum hydrocarbons or PCBs or, whatever pollutant it is or pesticides that you're trying to get out of the soil.
What do you guys think of outdoor remediation? How long would that take if you were using that, and maybe in conjunction with hemp that you're going to throw away, or maybe just use as to feed for the next year crop? Is that feasible within a year?
I'm probably not qualified to answer that question, but yeah, it just probably can be done if you're doing it pretty intensively, but utilizing biological diversity and. But I mean, I think generally if you're trying to do in situ bioremediation, you really want to be setting aside a number of years for that plot, because to be sure you're going to remediate the soil within 1 year is probably going to require some pretty hands-on and intensive techniques.
All right, this question is from Ron W. Is diversity in rhizosphere a consequence of our culture of hemp? If we fed compost tea extract, would there be more diversity?
I'd say probably yes. The
question to clarify, a compost extract is you just taking your compost and extracting it, whereas the compost tea is you're brewing it. So, based upon your assessment of your soil, if you're noticing, okay, I need more fungi or need more predators, you would then supplement that with inoculating with additional ones. Obviously, most times you're inoculating, you need more fungi, so you're adding more fungal foods to that tea. You would take that extract and brew it up. So yeah, you could supplement, but it depends upon looking at your situation, your soil from basically some microscopy, and then assessing, okay, do I need, what do I have the absence of? What do I need to kind of make up for? Predominantly it's going to be fungi or the predators of the fungi. Most oftentimes you're not going to be worrying about increasing bacterial diversity, but it's specific to your kind of what's going on in your bed and your root zone.
Yeah, I don't think it's a settled topic, but there's a lot of opinions that if you have a compost tea and it's very diverse in all of the soil food web groups, then it doesn't that's sufficient to add. Like, even if you're like trying to deal with a fungal deficiency, if your compost tea has a bunch of bacteria, but it also has a bunch of fungi, it's probably still good to apply, because the plants are ultimately the ecosystem engineers. They're going to feed the microbes. So the microbes that don't get fed by the plants are probably going to die off. So if you have your plants in there and then you're adding a big diverse array of microbes, and the plants are going to kind of select who they want to be there. So it's probably going to add to the diversity, I'd imagine.
Yeah, which is why like cover cropping, even in just conventional ag, is taking off, because the biggest issue is that they're leaving the soil just like bare. You know, they're not even, just when it goes fallow, it's just kind of a thatch layer. So if you put on any organism, but there's nothing to feed them, even in the quote unquote off season, you know, you're getting those microbial returns both in the active and passive of growing both your primary crop and then whatever's of cover that's going to be continuing to build that diversity in your soil and keep it going.
I will say as well, I learned from Dr. Lane Ingham that if you are making those, especially if you're going a little bit different with those compost teas, that brew those teas inside the grow area. I saw a lot more success when I was brewing it inside that grow room than outside the grow room. I hope that Dr. Lane Ingham could probably talk about that in the future, but I did see a night and day difference with that.
And think about like, what is like, you want to like, think about what is the ambient temperature of your growing environment, right? If you're growing, like, let's say you're growing like, you know, let's say like you're growing in the summertime, right? But you're brewing your compost tea inside with the air conditioning on, right? Those microbes are going to be, you know, growing in conditions that are a little cooler, and then you put them out in hot temperatures, not going to survive. So it's understanding like brew kind of the similar temperature conditions to like what that environment is going to be, the climate of where your plants are growing.
Yeah, I feel like that's a gold nugget for people that are just starting out on the tea journey. But I also will say that a lot of us that have been doing this for a long time, a lot of us feel like the extracts are now more time consuming and seem more beneficial. So take that for what it's worth. Julie D, are there any yeast or fungi that are not good for plants that you wouldn't want in your soil beds or pots?
Yeah, there are.
There are.
That's like the foundation of the field
of my we're gonna
basically the foundation and funding of the field of mycology was to study fungal pathogens and yeasts, and lots of them infect things. So yeah, there are a lot of bad ones, but when we talk about, you know, we were mentioning earlier the concept of like the beneficial yeast forming biofilms and preventing pathogens. And I know it's something that Elaine Ingham talks about a lot in terms of if you coat your plants in beneficial microbes, then even if there are pathogens in the environment floating around, they're probably not going to have an easy time infecting your plants because there's already like a strong community existing. But we don't want to like view fungi as being inherently good or inherently bad. They're just a very like diverse, complex realm of life that can be beneficial. There are good ones, bad ones. There's parasites, you know, they make things stronger, they kill things, they run the whole gamut. But generally kind of we find this theme that the more diverse the system is, the more likely pathogens will be suppressed and, you know, synergistic beneficial interactions will occur.
Definitely. And kind of an insight, kind of from talking from the soil food web thing, if you're doing microscopy, you're noticing, you know, hyphomycelium that's like less than 4 microns in diameter, so less than 4 cocci bacteria. It's probably more of like a opportunistic fungi, which can be a pathogenic in general, which kind of is showing a lower oxygen condition. And kind of on that note, kind of similar to natural farming, the whole reason why you add OHN to basically go on and make basically to go on and make IMO3 is the process that, you know, that OHN is that I believe the angelica root, the licorice, and the cinnamon, the garlic and the ginger are acting as basically like anti-fungal compounds for those opportunistic fungi. You're basically selecting for the fungi that, you know, if there's a niche, they'll hop in and fill it, you know, because they're able to survive as kind of in that certain community. So usually in environments where, you know, the best analogy, think about this, like think about like Mad Max, right? Like you get like people trying to survive, and you get like, you know, like you get like these bandits and raiders, and like things are scarce and there's not much community and biodiversity, versus you have like, this interdependence with all this biodiversity. There are niches and there's food and everything to go around, right? It's far less competitive because, something's exudate, something's waste can be another organism's food or bad process. So that's the whole thing about it. Yeah, there are species of pathogenics, but part of it too is a response to us kind of really blowing up the health of our soils, given the fact of overtilling, destroying that biodiversity as well. So it's the kind of perspective like the background of mycology, there are those species, but understanding the principles of that, what they're indicating. They're more of an indicator that they're there and making themselves known because there is a lack of balance in the system.
Chandler, how can you tell if the fungal hyphae is beneficial under the scope? You said something about a clasp or hook connection, but I didn't catch it.
Yeah, so what I was showing there is usually so in larger fungi like Basidiomycetes, Basidiomycota, those are the fungi that often do produce like the mushrooms, pretty much all gourmet mushrooms that we do eat, like the white rot fungi are Basidiomycetes. An indicative sign of a beneficial fungi of that size is they have this little nodule, this bump between their septa. It allows them to pretty much move the nuclei over as they branch. But an indicative of kind of identifying like the pathogenic fungi to the beneficial fungi is the diameter of their mycelium. Usually anything 4 microns in diameter above, that's 4 little bacteria as your reference frame point that Elaine talks about in the soil food web microscopy. That's an indicator of that aspect as well. And there's also kind of pigmentation and also the fact they're septated as well. There are some fungi that aren't septated. They're called choenic. They don't have septa, but usually the morphology of less filamentous and more kind of scraggly, a number of kind of molds or mildews kind of have that indicative, number of those mildews, for example, have an indicative kind of like a witch's broom kind of looking shape. And that's that mycelium and thin and transparent. Very, very goes to the more opportunistic one that are thriving in lower oxygen conditions.
Yeah, isn't the rule of thumb that I've heard is that if the hyphae is really skinny, like you were saying, and it's clear and it doesn't have a septa, then that means it might be a pathogen. It doesn't mean it is a pathogen, but on the converse, if the mycelium is pigmented, it's wide, it has septa in it, then it's more than likely beneficial. It's probably not a pathogen. But if you're seeing the skinny, clear fungi, those are more likely to be pathogenic, although I don't think you can be sure just from looking at it in a microscope.
Man, you guys are next level with a lot of the stuff that I guess we could see come to fruition, just literally have no idea why. So, all right, Clay W. What about mushrooms in beds? I'm thinking something easier like almond or snowcat.
You like portobellos or like
almond? Yeah, almond portobello is, I assume, what you're mentioning, and that can be mixed in beds too. I think it's just that's those are not white rot fungi, or those are more like tertiary decomposers that consume more like compost and manure and stuff like that. But if you're adding the right materials, I've never worked with them personally in that context, but I could see that they'd be beneficial to that as well. I don't know what you're referring to as the snow cap. I know there's like a
shaggy mane. He's referring to like some of those like shaggy mane or like anything that's going to be kind of more of like a compost or.
Yeah, I think any sort of fungus, any sort of decomposer fungus that can grow in soil on its own is worth experimenting with. I mentioned the Stropharia and oyster because those are the ones that I've personally seen used a lot. But yeah, I think any sort of soil fungus, like if it's a shaggy mane or portobellos, whatever, it's worth trying because it's the same concepts at play.
Yeah, the macro. If there's a macrofungi coming up in your bed, like a mushroom itself. That is not the type of fungus that's going to be like attacking the living plant. That's going to be degrading and decomposing, dead or like, moribund organic tissue. They're going to be the cyclers in general. Like, the ones we talked about previously, the pathogenic or decomposer ones. And even too, like there are some fungi that even show up in the plant that will infect the plant and wait until it dies. And then basically go about the process of decomposition. Like kind of like the first there strategy. Kind of holding it down up until the condition change in that plant material. But yeah, definitely kind of on that. Also, when it comes to with edible fungi, if you get mushrooms coming up out of your beds, don't eat them. Just ID them first. Definitely look at them and kind of take a look and figure them out. That's a great way to get into it. There's some great apps if you an app if you want to identify mushrooms called iNaturalist. It's like pretty sweet. It's like Pokemon Go, but for like taking pictures of leaves or mushrooms, plants or animals. But it's a great way to like snap a photo of something. It'll run the image through like an image processing algorithm, and it'll kind of give you the best guess, and then you can kind of key it out with other photos and kind of doing some Google searching as well. But yeah, definitely if you see mushrooms coming out, appreciate them, look at them, just don't eat them. You can touch them. Mushrooms, no, if you know, I think there's only one species of mushrooms that actually can subcutaneously be poisonous. It's like it's called the viral coral fungus. It's like in Australia and Japan. But most macrofungi, pretty much all macrofungi that are popping up out of your living soil beds, you can touch them. They're not gonna hurt you subcutaneously. Just if you're considering investigate them first, get a complete positive ID. I wouldn't recommend just kind of throwing them in your pan the minute you see them coming up. So.
You generally have to swallow a poisonous mushroom to get poisoned by it. Most of them you can actually put in your mouth, chew up, and spit out, and they won't poison you. Not that I'm recommending you do that, but just for the toxicity awareness.
Indeed.
And from like a terpene perspective, a full genetic profile perspective, I feel like when you start to see the fruiting bodies in your living soil, you need to know as a cannabis farmer that you're headed down the right direction, because that is when I feel like those start to fruit up, kind of like fall over and then continue the process, build that out. And if the air pod or the fabric pod is big enough, it seems like that those fungal areas just continue to build upon one another, and you'll start to see a variety of different fruiting bodies.
Mm-hmm.
Right. And diversity is key.
Yeah, absolutely. All right, John M. In leaf mold, black leaf mold indoors, how do you make sure you're not bringing other problems in with the wonderful fungus?
That's a good question. I mean, I think if your leaf mold is aerated and it stays like aerobic, I don't know, you might be bringing something in. I can't be certain of it. I've never observed adding leaf mold to something and then it getting infected with a pathogen. But then if you're bringing it inside, it's hard to say, but I'd say, once again, the smell test is a good one, because a lot of pathogenic molds will start to have, like when they get into your leaf mold or your mulch, they give off a certain odor. But yeah, it's hard to be sure what you're bringing in. But one benefit of leaf mold as a mulch source and a living mulch is that if it is just leaves, at least you're not going to be bringing in like seeds from weeds and things like that, which can happen with other types of compost. So that's one thing you probably don't have to worry about. But in terms of like, could some pathogenic mold be latently living inside the leaf mold, even though it's a bunch of other beneficial fungi, and then that one gets out of control when it goes into your indoor controlled environment because the other wild ones don't really survive there? It could happen. I can't give you a good answer to that question.
All right. I appreciate your time, guys, and I want to respect everybody else's time. So I want to make this like the last question or two. This one's an interesting one from my perspective, because I love Joe Rogan's podcast. But any comments on the mushroom mafia that Paul Stamets refused to talk about when interviewed by Joe Rogan?
Yeah, I'll say this. Back when I was doing mushroom farming, we had this older fellow who was a carpenter who helped build our grow room, our fruiting chamber. And, and he's just, older guy, lived around, he's definitely a worldly person. And now we brought this up about jokingly about, and then the Portobello Mafia, and he very dead serious was like, oh yeah, totally. Yeah. The mafia has been running the mushroom industry ever since it came about. So I don't know if this is true or not, but, I will just say this, that the button mushroom cultivation technique was discovered in Pennsylvania, Penn State University in the early 1900s, around the 1910s, maybe same time overlap as Prohibition, the rise of the Cosa Nostra, and, mushrooms are a big part of Italian food. And I've said too much already. I'll leave it there.
Yeah. Also the fine, the enterprise of carting materials. Most of the, Kennett Square came into being because, think about what we had before cars, horses. What do horses make a lot of? So most of the manure from Philadelphia was carted out to Kennett Square, and that was used as a viable substrate to start the Agaricus bosporus button mushroom cultivation industry.
I feel like it's silly, but the mafia probably has their hands in a lot of things, because I've always heard that olive oil is another one of those that they practically control.
And
they water down.
The whole plot of The Godfather, right? That's how Don Corleone, became big, was through importing olive oil from Italy.
Yeah, but sometimes with movies it could be, but apparently that's true, and everything's watered down, and they have a whole almost like monopoly on the real stuff. Yeah. Well, gentlemen, I appreciate your time. I'd love to be able to talk to you guys further. So let's please stay in touch. And, for you living soil nerds out there, how are they going to be able to contact you? Leif, I don't think you're, do you have social media or anything, sir?
No, I don't really. So you can email me at mycoscapes@gmail. I know it seems very counterproductive. I don't have a social media presence, but that's my email, mycoscapes@gmail.com. Or you can email me, or you can follow Craig on social media. So But,
I hope a lot of you do. Fantastic. You have a fantastic Instagram.
And definitely we're currently working on. We've recorded a bunch of episodes for this Applied Mycology podcast we're going to be launching. So we're trying to kind of create a platform, but also too like a regular scheduled show where we kind of cover details of like how to work with fungi, interviewing people that are like more in the DIY, research scientists, and also people that are interested in kind of intersection of application and understanding as well. So definitely look for that. We're going to be putting that out, probably the first episode in the next few weeks.
Excellent. Yeah, there's a lot of great podcasts out there. We also have one on Thursdays on the Future Cannabis Project. It's every Thursday, so please check that out. We put a lot of time and effort to those. So the people that we interview are definitely, we don't want to waste your time. So we know that your time is valued, just like today, where these gentlemen have taken the time to put together a fantastic presentation. So again, I thank you guys. Again, I want to thank Agro Coco for being the title sponsor. They're one of the main reasons why we're able to keep, not only pay our speakers, but also keep the cost down for you guys. So please check them out at agro.coco on Instagram. And again, I feel like a lot of us in the living soil world know about Blue Mats Sustainable Village. Shout out to Blue Mats. We appreciate all the love that you guys have supporting the SCCA. Until next time, guys, we'll talk further. Have a great evening.
Take care. Thanks for having us. Later.
All right. I think we're off, boys. Appreciate it.
All right. Can you guys hear me?
Yeah, we can hear you.
Yeah. Yeah. My video cut out the last minute. Thankfully, I was able to dial in. So.
Yeah, no problem. Just technology. I don't know, especially with a lot of these webcasts, webinars, all this stuff that's come out. I think they're trying to keep up with how many people are using it, but it seems to be that some companies are better than others.
Definitely. How'd you like the prez?
Fantastic. Honestly, it was.
Glad you liked it.
Well, I really like when presenters go out of their way to show visuals, to show certain key concepts. Mm-hmm. It's not everybody is digesting, especially at your level, gentlemen, and the whole, all of the speakers and presenters. A lot of us cannabis farmers aren't at that same academic level. So we do have to go back and watch these a couple times. And when you guys are able to give the amount of visuals that you gave in that presentation, I feel like cannabis farmers really resonate with that, and we're able to absorb a lot more of that information.
Hey guys, we did stop the recording, but we're still on with the audience here.
Okay, hey everyone.
All right, well, glad we didn't say anything ridiculous. All right, well, thank you guys.
Yeah.
Yeah,
right,
thanks.
Take care. Have a good one. Later.
Cool.
All right, Leif, Greg,
All
Daniel.
right. See ya.
See ya.